Good afternoon, everyone. It's a real pleasure to be here with all of you to chair this so interesting afternoon session of this interesting meeting. First of all, I would like to thank the organizers for this kind invitation. It's a real honor and a real pleasure to be here with all of you. Just to start and to be on time, I would like to present the first speaker. Is Professor Peter Bailey from the Botton-Champalimaud Pancreatic Cancer Center in Lisbon, Portugal. And Professor Bailey will talk about the genetic and non-genetic mechanisms driving progression and resistance to therapy in PDAC. After his talk, we'll have the Q&A time. Thank you very much, Professor Bailey.
Right, so I'd just like to thank the organising committee for inviting me here today to give a talk on some of the work we've been doing the last couple of years. And I'd also like to say that I'm not a professor, I'm just a run-of-the-mill PhD. So I think that's important to actually state straight up. Okay, have I got a way of pointing? So the majority of this work is published, but I think it's worthwhile to perhaps go over some of it and to point out some key aspects of the work, especially given that we're now entering into the age of KRAS inhibitor therapy. And I think there are some potentially interesting observations, at least with respect to genetic and non-genetic mechanisms underpinning pancreatic cancer that can play a particular role in KRAS inhibitor resistance. So I think we start probably at the beginning, and that really is what's been happening in the last decade. And really this is the foundational research that I've been a part of, even from the early days in the QCMG, Queensland Centre for Molecular Genomics, through to work that was completed at the University of Glasgow and onwards. And this work really focuses on two key areas, and that really is the genetic or genomic drivers of pancreatic cancer and the non-genetic or the transcriptomic subtypes of pancreatic cancer. And as you know, there have been a number of transcriptomic subtypes that have been defined for pancreatic cancer. They have a number of different flavours, and they've been designed and generated based on a number of different informatic tricks, if you will. But I think now the field is really centred on two broad subtypes of pancreatic cancer, classical basal, as you know. And of course, these subtypes were identified in resectable patient samples, and when we go to more advanced settings, such as metastatic setting, they tend to fall away a little bit. They tend to be a little bit more inconsistent, and we now have other subtypes, such as the Chanseng-Hui subtype, which tries to accommodate for this increased tumour heterogeneity. And I think what's becoming apparent to all of us is that, especially in advanced tumour, we're not looking at a single type of cancer, and in fact there's considerable intratumour heterogeneity. And pancreatic cancers tend to be found in a continuum. Now, of course, this is debatable, the starting and perhaps or the ending of this continuum. But nonetheless, there's a general consensus that we have classical and basal states, and also intermediate co-repressor states. And this plasticity, it seems, tends to be associated with the tumour microenvironment. So the tumour microenvironment can cause increased plasticity towards these basal-like states. And in fact, Jen Morton produced a very nice paper in mouse models where she ablated macrophages, and we saw a real shift in the neoplastic cell populations post ablation of macrophages. But chemotherapy itself is also known to affect this continuum, and ex vivo models have shown, cell models, organotypic models have shown that chemotherapy can also drive this shift towards basal-like subtypes. So to really look at the impact of chemotherapy in PDAC, we established a large cohort. This was in collaboration with John Neoptolemus at the University of Heidelberg, consisting of 171 specimens representing resectable patient samples that underwent adjuvant chemotherapy, primarily with gemcitabine, and locally advanced PDAC that underwent neoadjuvant chemotherapy. Those patients underwent, commonly were associated with treatment with neoadjuvant treatment with FOLFIRINOX, whoops, I'll go back, or Gemabraxane. And what we found from that study, I should point out that none of them had radiotherapy, and what we found in that study is that chemotherapy, at least neoadjuvant chemotherapy, could cause a very quite robust changes to both the tumour stroma and the neoplastic cell populations. I don't think this is particularly controversial, and other people have looked at this as well. But when we started to have a look at the established subtypes of pancreas cancer, we found that neoadjuvant chemotherapy enriched for the basal-like subtype. And we also found that the existing Moffitt subtypes were not prognostic in post-chemotherapy HDAC samples. So this actually goes against the grain slightly. We also found that high GATA6 expression, when we did our immunofluorescence, was associated with poorer outcomes in those patients that had received neoadjuvant chemotherapy. So to really understand what was going on, we performed multiplex IF using a number of different markers representing classical, basal, and intermediate states. And we found, interestingly, that there was a real shift towards GATA6, CYP3A, keratin 17 hybrid persister phenotypes in the post-chemotherapy patient samples. And here you can see them here, these GATA6, CYP3A, keratin 17 populations. These populations were found in the chemo-naive group, but really enriched post-chemotherapy, very significantly enriched post-chemotherapy. We also found that CYP3A, which is a cytochrome P450 member, was associated, at least in our organotypic models, with metabolism of irinotecan to more inactive versions. So as you know, irinotecan is broken down into SN38 in pancreatic cancer cells, and we found that SN38 wasn't produced at the same rate in these CYP3A positive cells, and we also found that any kind of inactive version or of the metabolite was secreted from the cells quite actively. But nonetheless, we're starting to see a real shift in the neoplastic cell populations post-chemotherapy. So this has really helped us to develop a model, I suspect, where we see intrinsic resistant mechanism where non-genetic priming or adaptation contributes to therapeutic resistance. And here we have these CYP3A positive classical hybrid cells that are then actually survive the first rounds of therapy and form a pool of persistent resistant cells that then go on to form recurrence. Now, I think this is interesting from the viewpoint of KRAS inhibitor therapy. There's now a push, and there's some clinical evidence to suggest that KRAS inhibitors are showing promise as a maintenance therapy in second line, and also that they may be useful in combination with neoadjuvant chemotherapy. But I think it's interesting that a number of studies so far have shown that KRAS inhibitors are enriched when you treat at least mouse models with KRAS inhibitors. You see an enrichment of the classical cell type post-therapy, cementing this idea that the classical states are associated with increased resistance to KRAS therapy. So we see also that there are basal-like states associated with better responses to KRAS inhibitors. This may be the case also post-chemotherapy. And also KRAS inhibitors in combination with chemotherapy increase efficacy and reduce metastasis to the lung. So there may be some specific role for this combination, but I think we need to be relatively careful about how we shape these studies in the context of this intratumour heterogeneity. Okay. The second line of work that we've been doing really is focused on genomic drivers of PDAC, and even back towards 2012, we started to see that the mutational landscape of pancreatic cancer is pretty heterogeneous. I mean, our hope was that we could identify actionable mutations for PDAC, such as other cancer types, and we found that we were relatively disappointed in that we only found a very, very small population of actionable mutations in DNA damage repair genes that were targetable with platinums or PARP inhibitor therapy. But now, of course, we see with the advent of next generation KRAS inhibitors that the majority of the tumours that we observe or find or may be targetable by a selective therapy. Okay. Although we often think about point mutations in pancreatic cancer, I think there's a growing, swell of evidence to suggest that as cancers progress, there are profound changes within the genome. And there's been some fantastic work by Scott Lowe's group where he's been looking at deterministic changes. And he's found that these changes are associated with disease progression and increased resistance. And there are three key characteristics of this increased or deterministic genomic evolution. One of those would be genomic instability or ploidy, an increase in major imbalances of KRAS, and also amplifications of a number of different oncogenes driving oncogene dosage heterogeneity. And all of these events conspire to increase progression, metastasis, and also resistance to therapy. We know that oncogene dosage variation has a very significant impact on cell states. Amplification of GATA6 is associated with the classical PDAC. Major imbalances of mutant KRAS are commonly associated in basal-like tumours, and intracellular heterogeneity of MYC amplification is associated with the more aggressive squamous-like tumours. Okay. MYC is an interesting beast, of course. So approximately 16% of PDACs are characterised by an exceptionally high MYC expression, whereas 13% have very low levels of MYC. And MYC's interesting because it's a major determinant of metastatic burden in advanced PDAC. We became particularly interested in MYC because of these focal amplifications. MYC amplifications tend to be associated with very focal events in genomes, and this seems to be associated again with metastatic burden. Why is that interesting? Well, focal amplifications are commonly associated with extrachromosomal DNA. So extrachromosomal DNA are a very interesting species of genomic elements that are gaining some interest in the field. And we looked at a number of studies, and in particular we looked at Kim et al., which was a large landscaping study of extrachromosomal DNA across multiple different cancers. And they found that 15% of PDAC comprised at least one extrachromosomal DNA. So this became particularly interesting for us to examine. So what are these extrachromosomal DNAs? Well, they are circular elements between 1 and 5 megabases. They are out with the chromosome within the nucleus, commonly associated with focal events. They're associated with increased chromatin accessibility and often occur as a consequence of a number of different chromosomal catastrophes, such as chromothripsis and breakage fusion bridge cycle events. These elements can also cycle back into the genome to form homogeneous staining regions and are very dynamic elements that can drive very considerable oncogene heterogeneity and impact tumour evolution. Okay. So to really look at the PDAC ecDNA landscape, we looked at chemo-naive post-chemotherapy and metastatic cohorts, again using the Heidelberg cohort, but also master HD cohort from Heidelberg, representing biopsy primary sites and metastatic material, and also a large panel of organoids from Verona, a very good collaborator of mine, Vincenzo Corbo, who you will probably know. And we looked for these extrachromosomal DNA using an informatic tool called Amplicon Architect. And what we found is 6 out of the 36 HD master samples comprised at least one ecDNA. 2 of the 22 post-chemotherapy samples are 9. So with 17 out of the 94 PDAC, at least 80% comprised a putative ecDNA, which is a relatively high number. There is some suggestion that ecDNAs accumulate post-chemotherapy, but we really haven't observed anything in this cohort. And also I should mention that this represents a non-validated set of samples and would require further validation. When we look at 41 organoids from primary chemo-naive patient samples, we find that, again, about 30% of the PDOs carry at least one extrachromosomal DNA. These ecDNAs were associated with biallelic inactivation of P53 and activation or amplification of CDK6 and CCND3. Most commonly, though, we found ecDNAs comprising MYC. We also had a number of interesting observations in that ARID mutations were highly enriched in the ecDNA negative cohort, suggesting that perhaps some of these chromatin modifiers could have a role in regulating the formation of extrachromosomal DNAs in, or at least maintaining them in cancer cells. We also performed a number of different validations, so that would be metaphase FISH, and here you can see quite clearly here chromosomes in blue and green is the ecDNA harboring MYC. You can see many hundreds of ecDNAs within the one nucleus, so they are quite apparent within the nucleus. You can see we've done circle-seq, which is another methodology, and validated them with capillary sequencing. Okay. What did we find? So we found that not all nuclei comprise a MYC ecDNA. We also found that these in interphases, we found that they cluster together or clump. This is quite a common characteristic of extrachromosomal DNAs. Extrachromosomal DNAs form these very active transcriptional hubs involving other parts of the genome to supercharge the expression of the oncogene. This is something that is of particular interest. We also find that there's an extremely wide distribution of MYC copy number within the same organoid. So this is driving this very increased heterogeneity within these cells. We also find that ecDNAs have a higher average MYC expression than linear amplifications of MYC. We also find a very wide distribution, again, of MYC expression in the same organoids. I think, very interestingly, we found that MYC expression does not scale. Oh, I'm going to get into trouble here. Yeah, I am. Yep. Whoop, doesn't scale linearly with copy number. So that means that even though you have lots of the ecDNA, it doesn't mean that you have an increase in the amount of MYC. And that's because the structure of these ecDNAs is very important. We see one of these organoids, which has a very, very high copy number for MYC, has a very, very low relative expression of MYC when you take copy number into consideration. And that is because the PVT1 promoter is absent in this organoid, VRO61, but retained in VRO10, which seems to be that there may be some competition between the promoter and the enhancers in this extrachromosomal DNA. There's some really interesting work coming out of the the grand challenge on ecDNA's dynamics in glioblastoma, suggesting that there's quite a lot of heterogeneity, structural heterogeneity in the extrachromosomal DNA pool, which can really affect the genomic evolution or evolution of these tumours. So we're dealing with a very complex beast that maybe is very, very difficult to treat. So very quickly, Vincenzo Corbo was interested in the idea of how the tumour microenvironment could affect extrachromosomal DNA. We all know that inflammation, for instance, can influence genomic events, but Vincenzo was particularly interested in Wnt niche independence. And forced Wnt expression of MYC induces niche independence in PDOs and provides an fitness advantage in the absence of these very important growth factors. The other thing about Wnt factors is they're associated with, or this independence is associated with malignant transformation of cells towards more mesenchymal cell populations. So Vincenzo was really interested in understanding what happens when we actually drive this Wnt independence in Wnt niche independence in these organoids that comprise an extrachromosomal DNA. And what he found was that he saw an increase in the ecDNA positive fraction when he'd actually induced the independence, an increase in mean copy number, an increased number of extrachromosomal molecules per cell, and an increase in MYC ecDNA copy number associated with morphological changes. So that's interesting. So when you actually have this population of extrachromosomal MYC organoids and you induce Wnt spondin or Wnt niche independence, you start to see this solid or cribriform morphology. Interestingly, when you actually do the opposite experiment, you put the Wnt spondin back onto the organoids, you see a shift towards lower ecDNA fraction and a change in the morphology back to this very glandular morphology. This doesn't occur actually in the ecDNA MYC negative organoid that has the amplification. So this seems to be driven specifically by the extrachromosomal DNA, which is of particular interest to Vincenzo. Another interesting aspect that we observed was that when we induce Wnt independence in these organoids, we see an increase in gamma H2AX foci in cells bearing high levels of ecDNA, but this wasn't observed when we just overexpressed MYC. So it's not a MYC-dependent process, but it's a process of the generation of more copies of the extrachromosomal DNA. So what does this mean? So there have been a number of different studies that have shown that ecDNA-positive cells can be targeted by CHEK1 inhibitors. This is a paper by Tang. This is the Michel group, a part of the ecDNA Grand Challenge, and they showed quite clearly that, they showed, I should say, not quite clearly, they showed that a new inhibitor, oral inhibitor for CHEK1, could selectively target these ecDNA-positive cells. They also found that in a gastric cancer model, that targeting with infragratinib alone causes resistance, but the combination with the CHEK1 inhibitor caused a synergistic effect. One thing that I mention here is ecDNAs as a vehicle of therapy resistance to standard cytotoxics and KRAS inhibitors. We know that from mouse models that MYC amplifications, focal MYC amplifications, are associated with increased resistance to KRAS inhibitors. There's also some studies in other cancer contexts where standard cytotoxics have been shown to, as I said before, increase the number of extrachromosomal DNA. But I think the jury's out. I think we need to obviously do more studies on understanding, how ecDNAs, especially those that comprise MYC, which is the common ecDNA in pancreas cancer, affects therapy resistance and tumour progression. With that, you can probably read more in that article that's published just recently. I'd just like to thank our collaborators, and in particular Vincenzo. That's Vincenzo there holding his small microscope. He's a really fantastic collaborator of ours, and he's the brains really behind all of the organotypic work. I'd just also like to thank the people at the Botton-Champalimaud Pancreatic Cancer Centre. We're just starting. This is what the lab looked like a year and a half ago, 2 years ago. We're now up and running with a number of different people. We have instruments on board. And I would also like to just introduce some of the members of our team who are working with us to develop a really comprehensive program for pancreatic cancer at the Botton-Champalimaud Pancreatic Cancer Centre. And then I would also like to, if you haven't registered, to invite you to our second meeting in October. So with that, leave it there. Thank you.
Thank you very much, Peter, for this very nice talk. Any questions from the audience? I don't see any questions. I would like to ask you one question. We are testing a c-Myc inhibitor now in a phase 1 clinical trial in different institutions in this country, and we are seeing very nice clinical responses with the combination of gem-nab-paclitaxel. Do you suggest another combination with this c-Myc inhibitor?
Gosh, that's a good question. Look, I wouldn't like to actually, I would like to say. I mean, it's not something that we've been actively looking at apart from understanding what's going on from a cell cycle checkpoint inhibitor. Some work that I did with the Heidelberg group, which I've now left, we were looking at standard cytotoxics and, again, these cell cycle checkpoint inhibitors. I think in general we found, and KRAS inhibitors as well, we found a general, to some chemotherapy, we found a general resistance to in organoids that comprised an extrachromosomal DNA that harbored MYC. We also found general resistance to KRAS inhibitors and a general resistance to some cell cycle checkpoint inhibitors. But then again, you know, I think you have to take some of those results with a bit of with a grain of salt. I think it requires greater understanding of the number of the number of ecDNAs or how the ecDNAs are being regulated to really get a handle on what's going on. So they were only preliminary studies, but the ecDNAs in particular I think, are probably a major vehicle for resistance. I know this isn't answering your question, but I think we've tried a number of different selective therapies, and you know, there's no, you know, one solution to the ecDNA problem. And you know, a lot of these CHEK1 inhibitors are being tried in the clinic and elsewhere. I think Jen Morton's tried them in mouse models and elsewhere with limited effect. These molecules tend to be more cytotoxic and not selective. So I think, again, you know, we must see the results from the grand challenge with a grain of salt. So I couldn't say one way or the other, unfortunately.
The microphone, please. Oh.
I apologize if the question is really silly. I just don't know whether the extrachromosomal MYC is only associated to pancreatic cancer after chemotherapy, or there is already existent,
so it
high existence.
It pre-exists the therapy. So you know, that's the thing. So I mean, we find in chemo-naive patient samples, you know, stage 2, stage 3. Okay, so it's a pre-existing aberration which people should consider in their work. I think these non-genetic and genetic mechanisms are interesting on their own. Obviously, cell populations, you know, we're dealing with a combination of the two in most tumours. Understanding the complexities of the tumours is something that we probably don't do enough of. Obviously, I know there are a lot of people here who work on mouse models. And of course, it's very difficult to really develop a therapeutic approach when the genomic and transcriptomic heterogeneity doesn't necessarily match with the patient cohorts that we inevitably treat. But anyway, that's just my 2 cents worth.
Another question?
Yeah, I would like to ask about RAS, whether it's detected in the cDNA, in the concept of resistance, but also in the more advanced tumors, that they're more unstable.
So we haven't observed KRAS ecDNAs in the cohorts that we've looked at for pancreatic cancer. In other cancer types, KRAS ecDNAs are observed. So we might hear something about that shortly. But fundamentally, yes, they can be associated with extrachromosomal DNA.
Okay. Thank you very much, Peter,
My
for
pleasure.
this excellent talk.
My pleasure.
Let's move to the second presentation. Thank you very much. The second presentation, we have the pleasure to present Dieter Sauer from the German Cancer Research Center and the Technical University of Munich, Germany. Dr. Sauer will talk about the tumor-host interaction in pancreatic cancer. Thank you very much.
Yeah, many thanks for the kind introduction. Many thanks to the organizers, to Carmen and Mariano, for setting up this fantastic meeting, and also for sharing this exciting data on the triple therapy, which I find super interesting and very promising. And it really shows that we are at a tipping point, and many future developments will be possible from now on, I think. We are very much interested, so we started, when I started to become interested in pancreatic cancer, which is now 25 years ago, we started about looking just at the tumor cell and at signaling pathways that might drive the tumors. But over the years, we realized that this is not all, of course, and that there's an environment and that there's an immune system which we have to consider. And therefore, we moved more and more into the tumor-host interactions. When we talk about pancreatic cancer, then many people say this is a very complex disease, which is of course true. We have many different cell types that interact with each other, but even in the century of single cell sequencing, we do not really well understand how they interact with each other and how they support the tumor. On the other hand, pancreatic cancer is also very heterogeneous. The most heterogeneous cancer type here in this study, which was done by spatial transcriptomics. This applies, of course, not only the tumor cells, which you see here, but also the stroma, which is very diverse, and the immune system. And finally, that's also important for therapy response and resistance, because the more complex the microenvironment is, the less the patients survive. This is quite surprising because in the end, genomically and genetically, PDAC is a very uniform disease. Many people already mentioned that. It's driven by KRAS and then a few tumor suppressors. We were interested in understanding better this diversity and how it evolves. And we used a model which has been shown here already several times. I just wanted to mention the people that developed these models, and that is Mariano and Carmen and Dave Tuvison from the States. And this really transformed the field because we had for the first time a model that really recapitulates pancreatic cancer in all its flavors, from the precursor lesions up to the metastatic state. And how useful it is, I think, has been shown today by Paco very nicely in the prevention. And Jen Morton showed that this reflects also therapy resistance. And these 3% of responders or trials that are positive is maybe also what we see in the human trials. So an excellent model, and we just activated oncogenic KRAS, and then you have to wait long because tumor evolution drives in tumor formation, and that takes up to 3 years, which is the lifespan of a mice, so this is in really late stage. And what we then analyzed, we analyzed these tumors, which are just KRAS driven, and what we identified, and this is this is the work of Roland Rath in Munich. So he identified that RAS amplification and allelic imbalance is one of the main drivers of heterogeneity of these tumors. And that is only possible if you lose tumor suppressor barriers. If you don't lose them and if you do not amplify KRAS, you get amplification of other oncogenes, we just have heard. At that time, we didn't consider ecDNA, but that's, of course, an important mechanism, as Peter showed. And that then finally determines a lot of the features of these tumors we see. So we see RAS expression is, of course, up in the amplified ones. These undergo EMT, so they have a very basal-like undifferentiated phenotype. Also in the histology, metastatic potential is high, survival is low. And we could show that also in humans. And recently, we could also show that this impacts a lot on the TME composition and also on drug sensitivity, and I will show you in the last slide one example for that. This is, of course, not all. So KRAS is not the only mechanism of heterogeneity. And therefore, what we did in the last 20 years in the end, so we combined a lot of different genes which we found in humans, mutated or altered, and knocked them out and combined them with oncogenic KRAS and so on. Then we generated a lot of cell lines out of these tumors. And this is now the transcriptional landscape of these. These are about 850 tumors. This is the UMAP of these cell lines, and you can see that this is a continuum in the end. We have two branches of very mesenchymal phenotypes. This one is here P53 associated. This is RAS amplified. And we have very epithelial tumors. And in the end, so this shows that it's, as I said, a continuum. In the end, it's a cloud, and we have extreme phenotypes, but we have many different flavors in between, which also maybe explains how difficult it is to treat these tumors. And what we did now with this cellular resource, so we tested then many inhibitors. We also tested the RAS inhibitors, the novel ones. And you can see we have, in the end, over the whole cohort, a 1,000-fold difference in IC50 values. So very, very sensitive ones, but also very resistant ones. And I think that reflects also what we see in the patients that respond. About 40–50% of the patients respond to the inhibitors and the others not. And you can also see here that this also depends a bit on the genotype, how well a cell responds to the RAS inhibitors. In addition to that, what we also have to consider is the subclonal architecture. So tumor evolution, of course, is ongoing. And what we observed, so now this is from just one tumor. We picked 40 clones. And you can see, this is now the copy number profile, that this is quite diverse. And we have some of these clones that have a MYC amplification or a chromosome 15 amplification where MYC is localized. Some have an RB1 deletion. And in addition to that, we have independent events of whole genome duplication. So it's not an initial event. We see that again and again, and it's not linked to the phylogenetic tree in the end. So all of that generates a lot of heterogeneity. And then now, since we have the clones, we can test them and see also the growth kinetics. And what we see, for example, that the diploid cells, they proliferate always faster than the others. So aneuploid cells are not as fit. However, they are more therapy resistant, as you know. I'm sorry for these transfer problems. They are more resistant, for example, to RAS inhibition. So also this genomic landscape impacts on the tumors. Now it's getting even worse. I'm very sorry for that. So that should show that we have a model where we can inducibly inactivate oncogenic KRAS genetically in established tumors by using a dual recombinase system, and by applying tamoxifen, we can knock out, so to say, permanently the driving oncogene. And what we then observe is, again, a lot of heterogeneity. So some of these tumors really are ablated. Others do not respond well to the ablation. Again, heterogeneity plays an important role here. So we have haploidy differences in these subclones. We have also very different morphologies, and you can see that also the copy number profile is incredibly diverse. And that impacts then also on therapy. We did single cell sequencing experiments where we use barcodes and then use the RAS inhibitor and then profile the cells over 21 days here. And you can see that one clone immediately outcompetes all others, which is a pre-existing primary resistant clone. And by looking then at the single cell data, we can see also in the transcription profile, so this is this clone 7.1, that this clone in the UMAP, in the transcriptional profile, doesn't change, whereas all others shift from this blue to this green state, which shows that we can already detect after 1 day of therapy the resistant clones. And that also applies for in vivo, so then we also implanted these clones in vivo, and you can see that we have always selection of certain clones and which are primarily resistant in the end to the therapy. So now my standard sentence is we always find a resistant clone and we always run into resistance, and therefore we need combination therapies, and I think that's still true. And Mariano and Carmen's data shows that very nicely, that with the triple therapy you can really eradicate that. But my usual sentence is then to say, yeah, okay, we have to target the whole ecosystem, we have to consider the immune system, and we need the help of the immune system to really eradicate these tumors. Now, since I've seen the data, I'm not so sure about that anymore, but for sure this triple combination therapy also impacts on the microenvironment in vivo. And we went very much now into the immune system and try to understand which kind of immune escape mechanisms are present in pancreatic cancer and how we might target that. And for that, we, of course, needed to profile or see first what's present in human patients. And we looked at a tissue microarray cohort of about 400 patients and looked specifically for neutrophils, for macrophages, and for T cells, because these were the main cell populations which we observed in our models, but also in human patients. And what we found that we see T-cell-rich tumors, we see macrophage-rich tumors, neutrophil-rich tumors, and also immune-excluded tumors. And we see in the end the same in the mouse. We have more immune-excluded tumors, but these are all end-stage tumors. So and these tumors here for humans, they are biased. These are surgically resected ones, so these are the more benign one or with the better prognosis. So I think this reflects more the end-stage situation. This reflects more the surgically resected, resectable fraction. And when we observed these findings that we have different immune microenvironments, it became clear that we have not the very best models at the moment, because we do not have models that really reflect these different immune subtypes which we observe, for example. And that's also then the reason why we do not understand what drives these different immune microenvironments. And we do not know, is this now a subclonal effect, for example, or is this, are these host states, and how the tumors escape the immune system. And things like contribution of cell plasticity and adaptation, we cannot really figure out. And therefore, I think it's clear that we need immunocompetent model that robustly recapitulate certain features in important subtypes of the disease. And we need spatial readouts in order to see the local microenvironment that is linked to specific clones as well. And therefore, we went now back to our cell culture resource, and we selected then 80 cell cultures based on the relevance, the genetic relevance, the transcriptional heterogeneity, also secretomic profiling of these tumors, and implanted these models into immunocompetent mice. And you can see that by doing that, we can recapitulate super high macrophage-rich tumors, which do not have neutrophils, but we have also tumors which have very high neutrophils, do not have a lot of macrophages. We can also recapitulate these T-cell-rich tumors. And these are really robust models where we can, again and again, test different therapies. Whereas when we have the KRAS only model, the GEM model, then we do not know what comes out in the end. If it's highly RAS amplified, then this is a very undifferentiated tumor, but we do not know that, and therefore we need much more cohorts, and of course also the timing is much more different. And what you also see is that some of these tumors are immunogenic, because if we implant them into immunodeficient mice, then the survival is shorter, but others seem to have induce no immune reaction to these tumors. By having now this kind of panel of different models for different TME subtypes, we can do quite some different things. So we can look at the TME and modes of immunosuppression. We are doing functional genetic screens. We can develop therapies, and we can also look at the impact of the microenvironment on these tumors. And I want to show you now just 2 examples of that. So what we see that there are different modes or principles of immune evasion of these tumors. We see in these neutrophil-rich and in the macrophage-rich tumors, there are, of course, very different cell populations within the tumor, but they all converge on T-cell dysfunction. And as you can see here by the transcriptional profile of this single-cell data. Based on the single-cell data, we generate a model where we predicted, so to say, what's going on. And this is also, we integrated secretomics data, also in vivo secretomics data here. And so the prediction was that CXCL5 attracts neutrophils, and then by APP signaling and MIF, they induce dysfunction of T cells. And on the other hand, and this is in the classical type and in the mesenchymal type, which is highly RAS amplified, we see that macrophages are recruited via CSF1, and then the immune reaction is blocked by PD-L1 signal. We wanted to know, is that now really true? So we knocked out these factors in the tumor cells, and we overexpressed them in the other subtype. And you can see here, so this is the neutrophil fraction, so about 2/3 of all cells are neutrophils, and if we knock out CXCL5, then we can reduce that quite dramatically. If we overexpress it in a mesenchymal line, then we can recruit neutrophils into the tumor. So CXCL5 seems to really play a role. And what we also observed is that when we block neutrophil infiltration, we get more T cells in the tumors. Or if we overexpress it, we get less immune cells into these tumors. So that was quite interesting. And next step was then that we wanted to know how are now these neutrophils reprogrammed towards T cell suppression. And we used here neutrophils from the bone marrow, so we isolated bone marrow stem cells, and we cultured them with conditioned medium, so without any cells and any cell contact. And thereby we can differentiate them into myeloid lineages, so into macrophages as well as neutrophils, depending on the subtype. And you can see here, for example, that these neutrophils are then long-lived. These are normal ones, they die within 4 days. If we treat them with conditioned mediums, they survive much longer. And they enter this immunosuppressive state with DC trail and PD-L1 high expression, which then blocks T cell function. We then profiled also these in vivo, the neutrophils, and you can see here that already in the blood, the neutrophil abundance goes highly up, and we see already primed neutrophils in the blood, whereas in the full immunosuppressive neutrophils are then only localized in the primary tumor. And what we then did is that we profiled the whole mouse, so to say, by single cell. So we profiled the bone marrow, the blood, the spleen, and the tumor. And this is the result here. And I go a bit further. This is the healthy mouse. This is the PDAC-bearing mouse. And what you can see is that the PDAC-bearing mice have a shortcut from the bone marrow that directly feeds them into this highly immunosuppressive population within the tumors. And that's shown also here. So these highly immunosuppressive neutrophils, they are only present in PDAC-bearing mice. In healthy mice, we do not have them. This is then the blood. What we then also observed is that this recruitment and the priming of the neutrophils by the tumor is already visible within the blood, and we can identify specific markers which show these recruited neutrophils, which enter already a kind of a pre-immunosuppressive state. But the final state is then always present within the primary tumor, where we have high CCL3 and high PD-L1 expression of these neutrophils. We are at the moment investigating what are the signals that, so what are the signals now within the tumor that push the neutrophils into this immunosuppressive state? We have some candidates, but this is not really robust data until now. Since we can recapitulate that in vitro with conditioned medium, we think that these are really secreted factors from the tumor cells. And maybe by blocking this mechanism would be a possibility to block immunosuppression of these tumors. On the other hand, we are of course also very much interested in these macrophage to have immunosuppression. If we knock out, for example, CSF1 in the tumor cells, then we can increase survival of this subtype. But animals then still die, but this is also only a perturbation that only affects the recruitment of the macrophages, and we have not applied now RAS inhibition or whatever on top. What is important is, maybe as a last note, is that the spatial localization of the macrophages are very important. So these are spatial transcriptomics data, and this is a tumor here. And you can see that these brown macrophages here, these are C1q, very immunosuppressive macrophages, are directly within the tumor, whereas others are kept, so to say, out. These are these SAA3 positive macrophages. And we have the feeling, because this is a therapy model, that these SSA3 are more anti-tumorigenic and antigen presentation macrophages. If we now look just at single cell data sets or at bulk data sets, then we cannot distinguish that and identify these functions. Finally, we used also these models to develop novel therapies, and that's published. Therefore, I just show the data in one slide. So what we observed is that a combination therapy of RAS downstream signaling and multi-kinase inhibition specifically targets the mesenchymal subtype. We get tumor shrinkage in these animals and sensitization of these tumors to checkpoint inhibition. And that's due to reprogramming of the secretome of the tumor cells, which are high in CSF1 and TGF-beta1. And that we can turn around. We can downregulate CSF1 and TGF-beta. And thereby we sensitize these tumors. Whereas in the classical subtypes, we have the opposite, which also shows how important it is to really target the right, or to give the right therapy to the right patient, so to say. Because here we make the whole thing worse, so we get an increase in CCL2 and CSF1, and we get even macrophage infiltration and more neutrophils into these tumors. With that, I'm finished. I would like to thank the people that did all the work. These are listed here, our collaborators, and of course, our funders, and I'm happy to take your questions. Thank you very much.
Thank you very much, Dr. Sauer. Any questions from the audience? Well, I would like to ask you a simple question that we have been targeting outside the tumor cells, the stroma, the immune system, but nowadays in the clinic, we don't have anything to target outside the tumor cell. And what we are doing wrong? I mean, how can we improve? Because you presented very nice data, very interesting data about the neutrophils that we have been paying a lot of attention in the lymphocytes, but not in the neutrophils. But what we are doing wrong, because we don't have anything to target outside the tumor cell nowadays for our patients.
Yeah, I think the stratification of the patient is not optimal at the moment. And there are studies, there was a Nature Communication paper last year that showed that HDAC inhibition plus checkpoint inhibition is efficient, but then only 3 patients responded very well. These were long-term responders, all others not. No one knows why these patients now respond. And I think we need really strong biomarkers for therapy stratification. And it also shows our combination therapy, it only works in one subtype. And we have to develop these therapies, I think, in preclinical models, and then try to translate them into the human and to the patients.
Any question? It is interesting that you presented very nice data about neutrophils, and we use neutrophils for a long period of time. It's a really old-fashioned risk factor for survival. I mean, when we see a patient with neutrophilia, high number of neutrophils at the first visit in the clinic, we know that this patient is going to have a poor prognosis, like patient with venous thromboembolism. It is interesting that we think we should target, and we should think about the neutrophils could be another target for our patients.
Yeah, I mean, I think it's a promising target. It's of course also a very difficult cell population. The survival of these cells is very short in vitro culture systems, and it's difficult to do, for example, genetic screens. But we have now developed some models where we think we can do that, and hopefully we will find some targets where we can really block these immunosuppressive functions.
If you don't have any questions, thank you very much. I think your message is very interesting. A good selection, good biomarkers to select, because we can improve our therapies. Thank you very much.
Thanks a lot. Thank you.
Well, it's a real pleasure to present Dr. Nuria Malats from Centro Nacional de Investigaciones Oncológicas CNIO from Madrid. Nuria is going to talk about the metabolic crossroads, exploring the diabetes, obesity, pancreatic cancer axis. Thank you very much, Nuria.
Thank you, Andres. Yeah. So thank you also very much, Carmen and Mariano, for your kind invitation to present here some of our results, and to Fundación Ramón Areces for your support to disseminate science over the time. So before I start with my talk, I want here to echo Yuval Dor's statements towards peace in our world. This is very important, I think, not only regarding life and regarding peace, but also for the freedom to do science. So he did his statements in a meeting in last May in Nice, and I want here to spread his words regarding towards life, peace, and science. So Probably it's not needed to show you that pancreatic cancer rates, both mortality and incidence, are increasing, have been increasing in the last decades, and they are still increasing. This has been shown regarding mortality in here in United States, but also in Europe in both men and women, and also for incidence and mortality. So it's very much needed to control pancreatic cancer crisis, okay? So how to control this increase in mortality and incidence? There are several levels of prevention, and you have realized that most of the talks today regarded actually to tertiary prevention, to treatment, and this actually is very much needed to have, here. So it's very much needed to have very good surgical resections and better treatments as the ones that you have shown here to reduce mortality. However, the proportion of patients that benefit of these treatments is quite small. So we have to increase this proportion of patients that could benefit of these treatments, and also to even increase more mortality, but also, and this is, this is secondary prevention, but also towards we have to invest more on primary prevention to also decrease incidence. And this is very much needed. So our group is mainly interested in secondary and primary prevention of pancreatic cancer towards what we call a personalized prevention of this disease. And to do that, it's crucial to need about which are the risk factors, which is the complexity of the etiology of this cancer, in order to define the interventions that we can apply to the population, not only the patients, but also the population. Paco has also shown a similar slide, and here it's a little bit more complex. This shows you the multifactorial etiology of pancreatic cancer. In the inner layer, you have, in gray, you have those factors that are well established associated with pancreatic cancer. So family history, chronic pancreatitis, diabetes, obesity, age for sure, tobacco, A, B, blood group, and alcohol increases the risk of pancreatic cancer, while allergy, asthma, atopic conditions decreases the risk of this cancer. In brackets and in white, you have the proportion of pancreatic cancer that could be prevented by avoiding each of these risk factors. So you can see that actually it's not a lot, okay? So probably we are missing a lot of these risk factors. In the outer layer, you have other risks factors or protecting factors in green that have been proposed, and probably they are involved, but the heterogeneity of the disease limits to have, it impairs to have them very well established associated with pancreatic cancer. Also, it's not only risk factors, environmental and lifestyle factors, but it's also genetic susceptibility. Right now, there's 40 plus 51 SNPs that have been identified associated with pancreatic cancer, explaining between 50 and 64% of the heterogeneity of the variation of the risk of pancreatic cancer. Microbiome has also been, it's also well established as a factor related, associated with pancreatic cancer risk, and the others, omics, epigenome, metabolome, immunome, regarding etiology, probably they are there, but still we need more studies to see how do they combine. It's not only that, it's not that on each of these layers of information, multimodal information, data is in contributing to pancreatic cancer, but it's the interaction and the correlation over the time among all of them. So this is super complex. scenario that we try to disentangle and try to characterize. In the group, we have contributed to that quite a lot, and we have done that with the resources of this study that it's called PanGene study that was conducted in 6 European countries. We included 2,500 cases, 1,500 controls during different time points from 28 centers of these studies, of these countries. And we have from all of these individuals very good epidemiological, clinical, follow-up, pathological information, and samples, biosamples, that then we use them to generate omics data. So genome data, genetic data, methylome in germline, microbiome, saliva stool, metabolome, serum, urine stool, and trace elements in toenails. So we are using these multimodal, and actually different layers of information in the same population to answer different questions that we have regarding etiology of pancreatic cancer and also other type of cancers. And today's talk regards to obesity, the crosstalk between obesity, diabetes, pancreatic cancer risk, and how these pieces of information what's the relationship between them, and how also genome, epigenome, methylome, microbiome, metabolome can also modify or interact or mediate the effect of obesity and diabetes regarding pancreatic cancer risk, so that we can place the pieces, the missing pieces of this puzzle in the correct way. So I start with very simple data results regarding the relationship between obesity, diabetes, and pancreatic cancer. So here, starting with type 2 diabetes mellitus, here you have the distribution of cases and controls from the study that I mentioned you before regarding those that self-reported not having type 2 diabetes and those that reported having type 2 diabetes. Here you can see that cases, almost 25% of cases reported having this condition, while only 13% of controls reported having type 2 diabetes. So this yielded a risk of 2.5 for those that reported having type 2 diabetes, that actually we could better classify them by using glycosylated hemoglobin. So those that reported having type 2 diabetes with the high levels of this biomarker have almost a risk of 4 for developing pancreatic cancer. What was interesting is that we observed that the risk was not linear according to the time since diagnosis of pancreatic cancer. And you can see here very clear. There's a peak of pancreatic cancer risk in the first 2 years before the diagnosis of pancreatic cancer with a risk of almost 6. And then the risk holds over time up to 20 years. It's significant. With a risk of almost 2. So these are known as 2 different types of diabetes, type 2 diabetes, the new onset diabetes and the long-standing diabetes. We could also better define and better characterize the risk of this new onset diabetes by using different biomarkers. With C-peptide, actually, that risk increases to 8.38. And not only that, we observe that men have even a higher risk of almost 10 in comparison to women with new onset diabetes mellitus. So that have a risk of around 3. So with this scenario, we wonder, what's the relationship? What happens here regarding the causality of long-standing diabetes mellitus with regarding pancreatic cancer? What's the role of BMI, obesity, and how it plays the new onset diabetes mellitus in this complex scenario? So this is our hypothesis, and we explore the causality using causal inference methodology, statistical methodology that we use in epidemiological studies. And this allows us to explore or to have at least a clue about whether a factor is causal or the relationship is causal or not. So we did that with Mendelian randomization, and this is the model that we use. And we not only consider here the type 2 diabetes mellitus alone, but also we had a second model using BMI, including BMI in the model. And here you have the results that we got. We explored the different potential, the different scenarios between the relationship between diabetes, long-standing new onset with pancreatic cancer, and but the most important results are the extreme ones. So pancreatic cancer actually is, we shown that because you can see that in both models, the single without BMI and by including BMI, the confidence intervals do not cross the risk of 1, so indicating that actually the relationship is causal here. So pancreatic cancer causes new onset diabetes mellitus. New onset diabetes mellitus is a consequence of pancreatic cancer. This was not shown before in epidemiological studies, and this we provided for the first time about this evidence. The other message that was important, very important, is that actually long-standing diabetes mellitus has not a causal link with pancreatic cancer, given that this is a very well-established risk factor for pancreatic cancer. So what happens there? How does it explained? No? We thought that the relationship between long-standing diabetes mellitus and obesity was quite complex also, and wonder whether they mediated, one mediated the effect of the other regarding pancreatic cancer risk. So we built two models. The first one on long-standing diabetes mellitus, and to see whether obesity mediates the effect of this condition, and then the other in the other way around. And what we observed actually is that both mediate the effect of the other. So increasing complexity in this scenario. So here is the challenge that we have, and how we see that. Actually, both are mediates the effect of the other, but none of them have a causal relationship with pancreatic cancer. So something else is before that, before those conditions. What is there, no? And that actually leads to these two conditions, obesity and diabetes mellitus, long-standing, and then finally leads to pancreatic cancer. So our hypotheses were that there could be different potential hypotheses here. The first one regarded that there's other lifestyle habits, diet, comorbidities, medication that could actually make the substrate a para-inflammatory substrate in the pancreas, and that finally could lead to obesity or long-standing diabetes mellitus. So we are exploring those with Harry and Brun. Brun is in the audience, and to see the complexities of all of the interactions between drugs, lifestyle, and other comorbidities there. The other hypothesis regarded to genome, and actually Paco mentioned you about the sub-inflammatory status that some of the genetic variants like NR5A2 or CTRB2 actually contributes to the pancreas, okay, without being very evident. But this could actually lead to this para-inflammatory, long-standing chronic status that finally explains the development of obesity and long-standing diabetes. The other is microbiome and metabolome, and we are exploring those. We have also contributed to that and have exploring those potential inflammatory related conditions in the pancreas. And we are now also analyzing how epigenome, the methylome, germline methylome, is also associated to that. But we wonder what's the role of each of these factors regarding pancreatic cancer risk, and how we have to include and and how we have to model each of them in our statistical models. No? So, and the potentiality here are 4 potential roles. One regards to be a confounder. You have heard about that several times. So confounder is associated with the outcome, pancreatic cancer, and the exposure. So, and here we are considering all of these exposures. And for example, diabetes is associated with pancreatic cancer and is associated with microbiome and with epigenome and also other risk factors. So it could act, diabetes, as a potential confounder, and we have to consider this and adjust our models for diabetes and obesity the same, actually. Another potential scenario is that these factors are colliders. If they are colliders, it means that they are consequence of both of the exposure and the outcome. But actually, long-standing and obesity are not a consequence of pancreatic cancer because of the time frame of the disease. So we excluded collider here. Mediators, it happens whether they are, as I showed you between the two of these risk factors, whether they are in the middle of the pathway between each of these exposure and the outcome of the disease. And we have to model this differently. And finally, the effect modifier, whether there's an interaction between the exposure and the outcome, so that with the presence of diabetes or obesity, the effect of each of these exposure actually increase or decrease the risk of the outcome. So we are exploring all of these roles regarding these exposures, and I will not show you all of these results because of the sake of time, but I will comment you on the genome and epigenome that we observed, and we saw that actually there's an effect modification between type 2 diabetes and this exposure regarding pancreatic cancer. So the first regards to the genome. We did a GWAS study with European population only, and we identified 53 variants in these loci that you can see here. This was done by Evangelina López de Maturana, who is also in the audience. And among those, we identified new variants of 2 genes that were already reported to be involved in genetic susceptibility of pancreatic cancer. So this is NR5A2 and CTRB2, and both of them have been commented by Paco before. So considering NR5A2 right now, what we have observed is that it was not only our new SNP that was associated with pancreatic cancer, but all the variation in the gene, considering the 12 previous SNPs identified plus ours here, using a scatter, we observed that actually the association with pancreatic cancer risk was highly significant. The whole gene variation is associated with pancreatic cancer risk. But very interestingly, we also observed that there was an interaction with diabetes, also in relation to pancreatic cancer risk, and the interaction was mainly, although borderline because of the sample size, but was mainly with long-standing diabetes mellitus. So there's an interaction between this exposure, the genetic susceptibility plus diabetes mellitus here in modifying the risk of pancreatic cancer. So we observed similar results regarding methylome here. What we did is with this sample size of cases and controls, we determined the CpG methylation level with the 850K of Illumina, Infinium Illumina. We applied these Bayesian reproducing kernel Hilbert spaces to observe how each of the layers that we consider here, that was the methylation, the main methylation layer, whether there was interaction with smoking, interaction with diabetes, and which type of diabetes. So what we observe here is that while the main methylation explained 15% of the risk, the variation in the risk of pancreatic cancer, when interacting with diabetes, it explained up to 33%. And it was mainly with long-standing diabetes mellitus. So here we also observe that there's something with these layers of information and actually on molecular markers that play a role, and it seems that may explain part of the risk of pancreatic cancer. What Evangelina did is to see with 100 top CpGs and 100 top SNPs, you have on the right-hand side, the genes, how do they overlap? These are CpGs and genes that are associated with pancreatic cancer in each of the subgroups. So non-diabetes in the bottom, long-standing diabetes, and new onset diabetes. And the same with genes. And what you can see that there's actually very little overlap of these genes and CpGs, or methylated genes and CpGs. So indicating that probably the pathways, the genetic pathways that are behind each of these population regarding the development of pancreatic cancer, they are different in these different subgroups. So we are exploring further all of these results in order to understand better and then to apply these in prevention of pancreatic cancer. So here is how very simple graphic to show how we have moved from very classical epidemiological models where we have started with an exposure, only long-standing diabetes mellitus or obesity regarding pancreatic cancer, considering also susceptibility factors that may modify this risk. So we move to integrative epidemiology, and we characterize the exposome with different layers of data, the genome similar with different data, and the phenome the same way. Actually, I haven't talked about the genome, but because you have already mentioned the heterogeneity of pancreatic cancer. So, and it may happen that different subtypes of pancreatic cancer have different etiology in this regard. So what we want and what we are doing actually is to integrate the different layers of information in the different compounds of this concept, this model, and then to model it all together. And we are using AI. We are experts. We have experts in the group that apply AI tools to integrate and to model the risk of all of that. So the take-home messages here is that, well, type 2 diabetes mellitus is associated with an increase of pancreatic cancer, but long-standing diabetes, while they have an increased risk, there's no causal relationship with pancreatic cancer. It acts as a mediator in the association between obesity and PDAC, and obesity in the same way acts as a mediator in this association. New onset diabetes mellitus is a consequence of PDAC, although there's some pleiotropic effects that were evidenced. And what is important is that new onset diabetes mellitus is highly associated with PDAC up to 2 years before the diagnosis, and it can be a marker to identify patients earlier that will have pancreatic cancer. The problem here is that we cannot identify and distinguish those that have or not have new onset versus long-standing. But the most important is that there's an intricate interplay of numerous factors that could influence the relationship between long-standing diabetes mellitus, obesity, and PDAC. And we need to disentangle all of this complexity here. And with that, I want to thank first the work that has been done by the people in my group. They are fantastic scientists that work very hard in this, not only in this study, but in others. All the contributors to PanGene studies, Paco, that you have seen, we have a close crosstalk, and we complement very much with our different angles to explore the prevention of pancreatic cancer and all the funding agencies. And thank you for your attention.
Thank you, Nuria, for this excellent talk. One question from the audience.
Mary Jane.
Hey, yeah, so super nice, like always. I have a naive question. It's surprising that long-standing diabetes, it's not causal for pancreatic cancer. And have you looked into patients, the ones that are in range, so that the glycemic levels are maintained in range, or the ones that are not? So meaning that the glucose levels are in range most of the time. And also some long-standing diabetics are insulin dependent at the end, or others are have oral medications. Have you looked into that in the range of the glycemia and in the medication they are taking?
Unfortunately, we don't have that data to look at and to answer that, but there's other studies. They are cohort studies that followed over the time the population, and they identify that actually those that are between the range, the normal range, they have a lower risk of pancreatic cancer. So if they respond to the treatment of diabetes, they have a lower risk than those that actually do not and have different peaks. Regarding insulin, we have observed that those treated with insulin have a higher risk than those treated with oral antidiabetes. So whether this is also a consequence of the aggressivity of the diabetes or not, we cannot say that, but still. No, we don't have that data, but we can think about, because we are exploring with Pablo, with a special transcriptome, this type of question. So maybe we can discuss on that later. Yeah.
One question?
I don't know if you can answer this. It's a naive question from a non-epidemiologist, but in the era of GLP agonists, is that going to help you remove or separate mediator effects, lifestyle versus diabetes, long-standing obesity, or is it going to make everything much messier?
Yeah, hope so. No, yeah. Still, no, there's no studies. Well, it says that the treatment with GLP-1, and antagonists, of inhibitors of GLP-1, they will help. It will help to reduce the risk of several cancers and several conditions, like not only obesity but also diabetes and others. Yeah, we are looking forward to seeing all of these results.
Thank you very much, Nuria, for excellent talk. Thank you very much. And let's move to the last talk of this session. I have the pleasure to present you Nelson Ducetti from the Cancer Research Center of Marseille, France. Dr. Ducetti will talk about exploring the molecular roots of resistance in pancreatic cancer. Thank you very much, Dr. Ducetti.
Thank you for the introduction. First of all, thank you to Mariano and Carmen for the invitation, all the staff for this beautiful meeting that have in the panel of speakers a so high proportion of friends and collaborators. So my work concern personalization of pancreatic cancer. And just a words about which is our strategy. Our strategy is to try to use the drugs we have right now that are not perfect, but that the ones that we are using right now in all the hospitals all around the world, gemcitabine and FOLFIRINOX, and trying to personalize the treatments and to use them in a rational way, taking into account the molecular biology of the tumors. So it's not needed to say that pancreatic cancer is very heterogeneous and we need markers or things that inform us how to treat the patients better. We have good drugs that work, but only a few percentage of patients will really respond, but all the patients will have the toxicity. The most important thing is to increase the better drugs we can, to look for better drugs, as Paco said this morning, and as Mariano said also, we have only gemcitabine and FOLFIRINOX, and how to choose between them. So we have some hope in targeted therapies to personalize with BRCA/NES, with MSI and fusions, and also with the arrival of the pan-RAS inhibitors or the RAS inhibitors that give hope. But even in those cases that represent, not for the RAS inhibitors, but for those target therapies represent a low percentage, and even for the RAS inhibitors, probably we will have resistance, and we'll need also prediction to personalize the treatments. So the strategy we are using, or we used, was to build a huge biobank of preclinical models. Why? Because when we began in 2010, working with patients where we know the response to the treatments, the results were really very tricky, very confusing, because we had the stroma and the other components, and overall, because if we want to work with biopsies that are taken with fine needle aspiration, the biopsies are really very small, contaminated with normal tissues that can come from the duodenum, from the stomach, or also infiltrates that really is very confusing. For the reason we use models that allow us to have very pure material, but lack completely the stromal component and the immune system. How to deal with this, I will explain you in the next minutes. The three models we used were primary cell cultures that come directly from the patients, organoids that are the three-dimensional in vitro cultures, and also the most classical PDACs. We had, what is important to say is that we had a huge cohort and containing not only resectable patients from resection, but also models that come from biopsies, from the primary tumor, or also from the liver metastases. With those models, we did what we named chemograms. So trying to challenge the model. Here you have the case for gemcitabine with primary cell cultures. So we challenged the models with different concentration of the chemotherapy, and we analyzed the percentage of viability 72 hours after the introduction of the drug. The other chemotyping we did was with PDX. You have here a resistant tumor the animal was treated with gemcitabine. The control and the not treated tumor grow in the same way, and here you have a very beautiful case of response with a relapse after the treatment during the time. The first model, very modern and very informative, are organoids that are also challenged with the chemotherapy, but we can do a 72 hours chemogram or follow the growth of the organoids by time-lapse video microscopy during the time. These kind of experiments inform us about the resistant models, the sensitive models, but also the ones that stop growing and represent much more chemostaticity than chemotoxicity. This was something that we used as an important information for the building of the predictive signatures that I will show you just before, just after. And with those chemograms, we built an anti-cancer drug chemosensitivity score, classifying the models that are sensitive and the models that are resistant. I say, well, the models, because the models are not coincident with the real history of the patients. In many cases, the patient responded well, and the model was resistant, overall in cells and in PDACs. That's maybe probably because we select some clones that are growing, and we use only the answer, only the molecular characterization of the models to build the prediction the transcriptomic signatures. So what we did was to use the models and compare and analyze which are the transcripts that are present in the sensitive compared to the resistant. Transcriptomic analyses were the most informative. Genomic differences do not inform at all. And other omics give us information like metylomics, epigenomics, but the problem is that they are much more difficult to translate to the clinics. And transcriptomic is the most easy way to go to the clinics. Then we said, okay, we will try to understand which is the best model, which is the model that is more, that give more information about the biology of the resistance and sensitivity. We compared the models by this selection, this selector that we named Pancreatic adenocarcinoma molecular gradient, that classify between pure classical, pure basal, and in a gradient way between classical and basal. And you have here the cells, the primary cell cultures, here the organoids, in yellow, the real patients, and in orange, the PDACs. You can see the models are very different, and something important is that all the models that are used here, we have the three models from the same patient, so PDACs, cells, and organoids from the same patients. The cells are mainly basal, the organoids are mainly classical, and the PDACs are very well spread, and is the model most similar to the patients. So what we did was to use the models in a complementary way, keeping the information that comes from each model about sensitivity and combining the markers with the others. For example, for primary cell cultures, inform us about the resistant phenotype and the basal-like kingdom, organoids about the classical kingdom, and PDACs was the most difficult model because it's very expensive and very long to have research. In this complementary way, we identify the transcriptome that characterizes the sensitivity from the models, and something important was to include the stroma and the immune infiltrate that is present in the real life, in the tumors. And we use a machine learning algorithm that train the signatures we obtain with the models to perform well in patients where we know the response to the treatment. So this was a cohort of about 100 patients where that was used as a training cohort. So the algorithm we obtained was named Pancreas View because it contained signatures for gemcitabine, for 5-FU, oxaliplatin, and irinotecan, the three components of FOLFIRINOX, but drug by drug. Abraxane is the signature is already done, but we are trying to validate it because it's not used. Abraxane is not well used in France because it's not refunded. And we are working with Teresa, with Tian, and with Florian to validate this signature. So I will speak today about the four signatures, gemcitabine alone and FOLFIRINOX. Something that we did was to train also the signatures to perform well in paraffin-embedded tissues and not in fresh tissue, because most of the hospitals are using paraffin-embedded tissues to do the pathology that we identify for the diagnosis. And the other thing is that the signatures need to work really rapidly. So 2 weeks was the maximum time that we hope. And in reality, we need only 3 days to run the algorithm and to prepare the RNA and the sequencing. But we need more than 1 week to have the block, the paraffin block, in the platform of sequencing. The preclinical validation of the signatures was done in the first time in 493 patients, all resected, randomized in 2 arms, one receiving FOLFIRINOX and the other receiving gemcitabine, is the PRODIGE 24 cohort, and the results are there. You can see in green the patients that are detected sensitive for gem and treated with gem, and here the patients that are detected resistant and treated also with gem. This result is quite good, but the most important thing is to compare to FOLFIRINOX. And when we compare with FOLFIRINOX, we observe that if the patient is detected sensitive to gem or not, the response to FOLFIRINOX is the same, so the signature for gem is specific for gem. And the other more important thing is that the patients that are treated with gem, if they are sensitive, they will respond as well as the patients that are treated with FOLFIRINOX, with less toxicity. Next step, to work with FOLFIRINOX. Here you have the signature for 5-FU, irinotecan, and oxaliplatin. You see there the sensitive and the resistant treated with FOLFIRINOX. And here you have the ones that are treated with gem, but detected sensitive or not for 5-FU, irinotecan, or oxaliplatin. That seems to indicate that the signatures are also efficient in the components of FOLFIRINOX. But now we need to see what happens if the patient is sensitive to 0 of the 3 signatures, to 1, to 2, or 3. This kind of analysis of interaction show that if the signature seems to be specific for each drug, and more signatures are present sensitive for the patients, more the patients will respond to the combo FOLFIRINOX. I need to say that in my first presentation, I say this kind of selection of drugs, and Alfredo Carrato was present, and he said to me, Nelson, I'm using FOLFIRINOX, not the drugs isolated. And we need, in this case, to binarize the signatures, and that's what we did. So we binarized the signatures. 0 or 1 is resistant, because you can see the statistical analysis that show only 1 signature sensitive is not enough, and 2 or 3 are sensitive. And we go ahead with the validation in patients. And maybe this is the most important result I will show you today. You have here in green, or in blue, the patients that are sensitive and treated in agreement with the signatures. The green are for FOLFIRINOX and the blue for gemcitabine. And here you have the violet that are the patients treated in the opposite way to the prediction. So this is important because it represents 1/4 of the patients that can be treated in the correct way. And we can increase the expectancy of survival and overall avoiding toxicity if they are sensitive to gem. But maybe the most important result for the future are the red one, that represent 27% of the patients that are resistant to all. When I showed this result to Paco Real, we were in his office, I remember very well, he said, Nelson, those red patients, those red blood patients are the ones that must be included in clinical trials if your signatures are really prospectively validated. And the other thing is that they can inform you about the biology of the multi-resistant patients. Because in this case, if we took only the patients, the red line patients, and we avoid to have the long, short survival violet, we can have a specific information about the transcriptomic characteristic of those multi-resistant patients. So those signatures were also validated in metastatic patients. The results were published. The results are very similar. And 3 clinical trials are right now trying to validate in a prospective way the signatures for GEM, for example, for the GEM and FOLFIRINOX signatures in Paxine at Curie Institute with Cindy Nussier for metastatic patients that are fit for FOLFIRINOX. GEMSign in the Institut Paul Calmette in Marseille by Emmanuel Mitry, that will test only the GEMSign signature because those are metastatic patients that are not fit for FOLFIRINOX. And the first is a NeoPredict clinical trial that is led by Brice Chanès. It's a national trial and will consider the borderline resectable or advanced patients that will be treated with neoadjuvant chemotherapy. For the future, the multi-resistant patients, of course, when I come back to the lab, I discussed with Nico Fraunhofer, he's bioinformatician, he's working in the cohorts, and we analyzed the multi-resistant patients versus sensitive in resectable and in metastatic, and we identify many, a signature, maybe a signature or a component that contain all the transcripts that are upregulated or downregulated in those patients that are multi-resistant. This signature is not really a predictive signature because it's not done to predict, but more overall to inform about the biology of those tumors. The most effrayable thing, the worst thing that we observe is that those multi-resistant cells, if we go deep into the intratumoral heterogeneity, are present in all the patients. Even in the patients that are the most sensitive, they contain some proportion of cells that are multi-resistant. So that means that we can have a good response, but then you have a relapse, and the relapse will increase the proportion or the ability of the cells to resist to the treatment. But, as I said, this first signature is not a predictive signature, it's an informative signature. To do a prediction, we prefer to work with single-cell analysis to go deep into the heterogeneity of the tumors and to identify the different proportion of the cells that can be, for the first time, multi-resistant, and at the end, maybe, the different proportion of sensitive cells in a tumor to prepare combos in a rational way. So to do single cell, you all know that we need to have a good single cell reference. This is the most important thing. Single cell reference, because it's this single cell reference that will allow, by deconvolution, to identify what happened really in a patient from a biopsy and from a bulk RNA-seq. The single cell reference in most of the cases is done with single cells that come from the patients, and where we can work with 10 millions of reads going not so deep into the transcriptomic information. It's like we are only seeing the top of the mountains, even maybe the tip of the mountains, because it's really the very highly expressed genes that we can detect in this kind of single cell analysis. We try, and this was not so informative because we do not find the same markers that we find in the patients in bulk analysis in the single cell analysis we were able to do. So our strategy was to use the 45 primary cell cultures we have because they are really homogeneous. It's like a clone. And we can do a 100 million transcriptome, very, very deep, and consider that this like a single cell because they are clonal. When we compare these 44 primary cell cultures that come from different patients, we observe that there are 4 clusters, and then we go to see if in those clusters we can find the markers that indicates multi-resistance. Those clusters were, the G1 cluster was classical, and all the other clusters were basal. That's normal because the primary cell cultures are mainly basal, but we have one cluster that is classical, sorry. Then we go for the analysis more deep to see if the markers of multi-resistant are present. They are present in 3, and the EMT markers are present in 2 of the clusters. But pay attention, so the cluster G1 is the one that is the most sensitive. We have 4 other clusters that are intermediary, and the most important is the pink cluster, that is EMT, basal-like, and presenting highly proportion of markers of multi-resistance. As I said to you, even the patients that are more sensitive contain all the time cells that are multi-resistant that are from the pink cluster, that is the more aggressive. Then we observe also that the presence of the pink cluster increase between resectable borderline metastatic from the primary biopsy and metastatic from the liver metastasis. And the blue, the more classical, more sensitive, decrease in the same gradient of pseudo-time. Because we can consider that the resectable patient is somewhere before to the liver metastasis. Same thing after the treatment, as we see this morning, treatment modify completely the ecosystem of the tumor. And again, we observe an increase in the aggressive cells and a decrease in the sensitive cells. Finally, to go to the patients, we used this single cell analysis by deconvolution to identify what happened in the patients. You will see, so here is again the PRODIGE 24 cohort, about 400 patients that were treated with GEM and with FOLFIRINOX. You can see here the different clusters we obtain. The pink one is here. And when the proportion of the pink cluster is high and the blue cluster is low, the patients survive less than when the proportion of the blue cluster is high and the pink is low. This is a prognostic signature by the convolution, by analyzing the transcriptome of a single tumor, so inside the tumor. But this is a proof of concept that transcriptome can inform about the proportion of cells that can be multi-resistant, and maybe in the future we will also be able to identify the sensitive cells that are the more interesting. So helping us with this kind of strategy, we identify also some markers, some targets that can be potential targets for the future. And overall, we identify phenotypes that are multi-resistant and mechanisms that are multi-resistant. What we think is that most of the markers that come up are basal markers, and overall markers that indicate squamousness and capacity of the cells to tolerate the treatment. That means that there is a huge response to stress. Many stress genes are expressed during the time that to allow the cells to recover and to come back again. Then when the treatment stops, to come back again to the normal phenotype. But the ecosystem, the global ecosystem of the tumor will be able much more rapidly to respond to the treatment the next time we will come back again with the chemotherapy. So the signature was named Panc Profiler, is for the moment a prognostic signature. In the future, we would like to include the microenvironment, because here we have only the response from the epithelial cells, and we know very well that the tumor is an ecosystem where stroma and immune infiltrate is extremely important for the response. And the next step is to validate the targets. So the take-home message is that transcriptomics can inform not only for the prognosis, but only for the response to the treatment, that if we have a good signature for gemcitabine and the patients are sensitive to it, they can be treated with gem with less toxicity than FOLFIRINOX. That we have Pancreas View that is able to perform not only in resectable patients, but also in metastatic patients from biopsy from the primary tumor and metastatic sites. And we think that integration of single-cell transcriptomic and deconvolution is, again, something very important for the future to analyze the intratumoral heterogeneity. Just 30 seconds to acknowledge all the patients that allow us to go ahead with this kind of studies in the retrospective and in prospective clinical trials. Emmanuel Mitre and Brice Chanès from the Institut Paoli-Calmette, other hospital in Marseille, Rémi Nicole and overall Nico Fraunhofer, who are bioinformaticians and are developing the signatures. Rémi for the first round of signatures, Jean-Pred and the others, and Nico Fraunhofer for Pancreas View, the new one, the newest signature we have. Cindy Nussier, that is conducting the clinical trial in Curie, and the other collaborators in France, and for the PACA team, the pancreatic cancer team, led by Juan Giovanna and myself, and that is placed in the university campus of Lumini in Marseille. Thank you for your attention.
Thank you very much, Dr. Dussetti, for this very interesting and provocative talk. Any questions from the audience?
Thank you very much, Nelson. Excellent presentation and very informative. Have you already introduced or plan to introduce the variable of splice variance to enhance the prediction power of your algorithms?
Could be an excellent opportunity. I think it's very complex for me. So I'm trying to collaborate with you, with Claudio Sette also, and to help you in the way to could incorporate in our algorithms this kind of complexity, because for the moment we are really very with our foot on the floor and trying to have an answer very rapidly. We have at least me, I have a feeling of urgency. Trying to discover new drugs is very important, but how to treat the patients the next month? This is why we are trying the easiest way, the more practical way, the cheapest way to treat the patients next month with gemcitabine and FOLFIRINOX, even if they are not so new.
Thank you.
If there are no more questions, we are run out of time. Oh, one question?
Yeah, I don't need to say again that I will do it, that I think that yours are the best signatures so far. But I have a question.
Yeah.
One of the challenges in pancreatic cancer, which is not so much of a challenge in other tumors, is when you're using a surgical specimen to define which region you're going to select for macrodissection for signature analysis. I mean, just because the tumor infiltrates, there's not an invasive front as in colon cancer or so. So it's very complex. There's a lot of stroma and a lot of chronic pancreatitis resulting from the tumor that integrates with the tumoral glands. So have you ever taken a number of blocks that have been, where the region has been selected by a pathologist and given them blindly to another pathologist and see how much difference is there in the classification of the signatures in the 2 or 3 different pathology-selected regions?
Not yet, Paco. It's an excellent question that we are trying to answer with Jérôme Cros, who has, for example, he has different punch in different regions of the tumor to see if the signature all the time predicts the same thing. The PRODIGE 24 cohort was selected by Jérôme, and he selected the region of the tumor that contained more than 70% of cellularity, so more than 70% of neoplastic cells. There is the cohort where the signatures perform the best. With biopsies, about half of the biopsies right now are not transcriptomically contributive. So that means that half of the signatures do not contain enough cells to be useful for the prediction. So this is a real problem. Another thing is that many times biopsies, because they are taken many times in different parts of the tumor, are more informative of the globality of the tumor than the resection. Where in the resection we will punch really only one part of the tumor, even though the signatures done in only a punch are predicting pretty well. So we think we can improve the signatures. This is the version for FOLFIRINOX, the Pancreas View version 1. Maybe with the time we will do like iPhone, 1, 2, 3, 14, 15, 16, and improving the signatures in the future with the splicing and with this kind of heterogeneity that we can have.
Thank you very much, Professor. It's a real pleasure. Yes. Okay, we are run out of time. I think we have the break. I'll see you in, I would say, in 20 minutes, 10 to 5. And thank you very much for all the speakers for this very interesting and provocative session. Thank you very much.
Okay, in the sake of time, because we were supposed to start at 16:45, and now it's 16:56, we're going to start the last session. We're going to switch the second for the first talk, because Laura has to take a train, and she's going to be the first speaker. She has titled her talk, "The Long and Winding Journey to Inhibit an Undruggable Target in Pancreatic Cancer and Beyond." It's like poetry, but it takes a lot of time to be a good researcher like Laura, that is coming from the Valle d'Hebron Institute of Technology. When you want, you're welcome, Laura.
Thank you very much. So, It's a real pleasure to be here today, and I would like to tell you about our progress in developing, finally, a MYC inhibitor for clinical use. So my journey started from the very beginning, since I was a student working on MYC. MYC is a very peculiar oncogene. We already heard several things about it, but one thing that I would like to remind you is that MYC is the most deregulated oncogene in human cancer. And I use the term deregulated on purpose, because normally we work on oncogenes that are mutated in cancer, assuming that they would be the real drivers of tumorigenesis. And in this context, MYC really makes an exception, because it's not usually mutated itself. It's actually induced by the other mutations that we find in cancer. So, for example, the usual suspects like KRAS or EGFR or PI3 kinase, they all use MYC inside the nuclei as their main transcription factor that then orchestrates the transcriptional programs that allow cancer cells to thrive. So MYC is responsible for promoting proliferation, but also supporting the tumor microenvironment, for example, building new blood vessels. And last but not least, MYC has been related to immune suppression. So cancer cells that have high MYC also produce PD-L1 or CD47, so all these signals that say, don't eat me, don't see me, to our immune system. Because of all these reasons, we always thought that inhibiting MYC would be really an important step in our fight against cancer. But unfortunately, for a very long time, MYC has been considered an undruggable target. And to date, there is no approved MYC inhibitor in the clinic. Just to summarize for you the reasons why MYC has been considered undruggable for so long, the main reason is a technical reason. MYC is an intrinsically disordered protein. What does that mean? That it's a protein that it looks like spaghetti. Allow me, I'm Italian, so I can say that. It means that when you put spaghetti on a plate, they fold every single time in a different way, right? So this is what happens with MYC. You can find it folded in the cell in so many different ways that it's really difficult to inhibit it with a small molecules, because finding a small molecule that recognizes it specifically is really, really hard. It's like designing a key for a lock that changes shape all the time. The other reasons are the following. MYC is a nuclear protein, and not all drugs get to the nuclei. It doesn't have an active site like an enzyme can have. And there is a MYC family that comprises c-MYC and MYC and L-MYC. And they can replace each other in some situations, so we better inhibit them all. And last but not least, for a very long time, it was thought that inhibiting MYC would cause catastrophic side effects in the maintenance of normal tissues. But as a student, as a naive student, I thought that nobody had tried really for fear of these catastrophic side effects. So I decided to test this. And I started from the basics. So MYC, in order to function, needs to dimerize, to form dimers with this protein called MAX. And MYC and MAX together bind the DNA like this. When they do it, they form this very defined structure. It looks like a forceps, like a clothes peg attached to the DNA, as you can see. And this is our opportunity to inhibit it, because it has a proper structure. It's not intrinsically disordered anymore at this point. So what I did was to design a fake MAX, an alternative partner for MYC. And I called it HomoMYC. So HomoMYC is this protein domain of 91 amino acids. It's based on the sequence of c-MYC, but with 4 amino acidic mutations in the leucine zipper. Because of these mutations, while MYC can only dimerize with MAX, HomoMYC can do much more. First of all, HomoMYC can dimerize with MYC and sequester it in dimers that cannot bind the DNA anymore. At the same time, HomoMYC can form homodimers and heterodimers with MAX that instead bind the DNA, but they are inactive protein complexes, so they shut down the transcription of the target genes. That means that we have a double mechanism of action here: the active sequestration of MYC away from the DNA and the occupancy of the DNA with inactive protein complexes. HomoMYC has been validated by us and by many other groups around the world as a pan-MYC inhibitor, because it can do this with c-MYC and MYC and L-MYC. And it was used mainly in its transgenic form in mouse models of cancer, where it showed efficacy in all of them, independently on the driving lesions that these tumors had, or even the tissue in which the tumors were, reinforcing the notion that MYC is an obligate conduit for different mutations in cancer. This model also helped us to establish that all the catastrophic side effects that everybody was expecting in normal tissues didn't happen. Normal proliferating tissues simply slowed down, but they didn't suffer at all. So there was an excellent therapeutic window associated to an extraordinary therapeutic impact. But even so, this was just a transgene mouse models, and we wanted to get to patients. And at that time, a lot of reviews were coming out. These are just sentences taken from reviews that said that OMOMIC was a molecule too big and bulky to be directly delivered to cells. It's 91 amino acids. It's smaller than an antibody. And OMOMIC is essentially just a proof of concept and can only work as gene therapy. Once again, nobody had tried. So there was this very interesting paper from Pierre Lavigne in Canada that showed that the max VHLH zip domain could penetrate cells and have some anti-MYC activity. Of course, max and OMOMIC are very similar, both in sequence and in structure. So if max could be directly delivered to cells, maybe OMOMIC could do it too. So I had this fantastic intuition, and I hired somebody from Pierre Lavigne's lab that started producing OMOMIC as a purified protein. This is Marie-Ève Beaulieu, that started as a postdoc in my lab, and started producing OMOMIC as a recombinant protein in bacteria. We started using OMOMIC in cells, and surprise, surprise, OMOMIC was able to spontaneously penetrate cells and reach their nuclei. This is an image taken 20 minutes after adding OMOMIC to the medium. So this was really unexpected, that OMOMIC by itself could penetrate cells spontaneously and be used as a drug. So with this finding, we were able to patent the use of OMOMIC in 2013 as a cell-penetrating protein and be used in medicine and in cancer. With that, we basically founded Peptomic with the first patent protecting this. And with Marie-Ève, we were the first founders, together with VHIO and ICREA, of this company that was supposed to bring OMOMIC to the clinic. So, of course, we had to show that this purified mini-protein could do exactly the same as the transgene had done. And this was the first publication in 2019 in which we showed that the mini-protein could be given intranasally or intravenously to mice. I don't want to spend too much time on this, but I want to show you some of the results that we had in this paper. First of all, when we suggested to use OMOMIC in the bloodstream of mice, people told us that peptides get degraded very quickly in the bloodstream, approximately 20 minutes. So it wasn't a good idea. So we started thinking that we had to do topic administration. In this case, we were interested in treating lung tumors. So we thought that we could give OMOMIC intranasally. And that's how we started. This is OMOMIC given, this is fluorescent MYC given intranasally. And 1 hour after the intranasal administration, all the lung is basically positive for OMOMIC. But to our surprise, also other organs were positive: lung, liver, pancreas, kidney, intestine. This already suggested that through the nose, OMOMIC had traveled through the bloodstream and reached these other organs. So it had survived the bloodstream. But let's stop for the moment on the lung. So we looked at radioactive OMOMIC in the lung. And in this case, what we saw is that 24 hours after the intranasal administration, OMOMIC has accumulated in the lung tumors. This was the basis for another patent protecting the use of OMOMIC as a theragnostic to identify where lung tumors are. So OMOMIC is retained by lung tumors and washed out from the rest of the normal tissue. This was really interesting to us, and again, serendipity. So we started treating animals that had adenocarcinoma in their lungs, and here you can see some longitudinal studies done with it. And in this case, you can see the mice treated. So this is the onset, and this is the end. Oh, well, you can more or less guess what I'm pointing at. So the onset and the end point of the experiment. So this is a 4-week experiment, and you can see that in the vehicle-treated animals, there is growth. Let me try again. There is growth of the tumors here, while in the omomic-treated animals, there was no significant growth of the tumors at all. This, to us, was simply a sign that we were having a cytostatic effect. But when we looked at the pathology report, the pathologist told us that the animals treated with the vehicle had, as expected, mostly adenocarcinomas. But the omomic-treated animals had regressed to having more than 50% of tumors that were represented by either adenomas or hyperplasia, which is not cancer anymore, right? So this was really encouraging, and it was very interesting to us. But when we went to the regulatory agency to propose a drug given intranasally in oncology, that didn't fly. They told us that we needed an intravenous drug. So we revisited the possibility of giving omomic intravenously this time, right? And hoping that this would work, we started administering omomic once a week intravenously, 30 milligrams per kilo. And we found out that actually the half-life wasn't 20 minutes, but approximately 49 hours in the bloodstream. So this mini-protein was really resistant to degradation. This is just a xenograft model in which we showed that this very, very resistant type of tumor that was EGFR mutant, PI3 kinase mutant, P53 mutant, and resistant to erlotinib was instead responsive to omomic. The first response was slow down in growth, and by the 3rd, 4th week of treatment, the tumor started crushing. And this is something that we have seen more than once. So this was the very first proof of concept that this could be given also intravenously. Since then, we have published the same results in other type of tumors. This is an example of a study in breast cancer, in particular in metastasis of breast cancer, because it was the first time that we tried to see whether this would work on metastasis as well, and we got positive results. But of course, the most important thing for us was to bring this to the clinic. And I'm very pleased to say that this got to phase 1 clinical trials in 2021. So, where do we start? We decided to go in all-comer solid tumors. MYC has a role in the majority of human cancer. Let's try them all, everything that we can do. So this is a classical phase 1 study, dose escalation, and we tested omomic once a week by intravenous administration in approximately 30 minutes. The first 2 doses were expected to be below the efficacy dose. So we got accelerated approval. So we got the permission to do only 1 patient in dose level 1 and only 1 patient in dose level 2. And then we switched to the 3+3 design, the classical one. Well, surprise. In dose level 2, we had a pancreatic ductal adenocarcinoma patient that stabilized. And we were very surprised. Again, it was sub-efficient dose. And we thought it was just by chance. So we went up, and we went to dose level 3. Another pancreatic ductal adenocarcinoma patient stabilized. And at that point, we started hoping. And then we saw somebody with a sarcoma stabilizing, salivary gland adenocarcinoma, and so on. So just at a glance, what we saw was that approximately half of the patients stabilized at their best response as a consequence of omomic treatment. I just want to underline a couple of notable cases. This is a salivary gland adenocarcinoma patient that stabilized for more than 2 years, 26 months. And this patient was in a very bad shape when he entered the trial. He had an open fistula on his face, and he was suffering a lot of side effects, of course. Then there was a sarcoma patient, the second one here, that was known as a bad responder to previous lines of treatment. The last 2 treatments had lasted less than 1 month for this patient. So he entered the trial with very little hope, and he stabilized for 8 months. And then there was this very interesting PDAC patient that I was mentioning before. This PDAC patient was a very interesting case because by RECIST criteria, he showed an 8% reduction in tumor size, 8%. But by circulating tumor DNA, he had a reduction of 83%. So we decided to look at this patient further, and we looked at it by radiomics, three-dimensional volumetric reconstruction of her tumors. And what we saw was that the three-dimensional reconstruction showed us that this patient had a 49% reduction of total tumor burden, including in liver metastasis. This was really a very nice signal that we saw, and it was really interesting. And I want to stress this difference between RECIST and three-dimensional reconstruction of the tumors because it's something that we are seeing again and again with pancreatic cancer. So the pancreatic cancer lesions are so irregular that assuming that they are spheric ones is very wrong, and it's an overestimation of their size, just to mention it. Anyway, pancreatic cancer was our champion, and we decided to go ahead with it, and I'll get back to this in a minute. We also showed by single-cell sequencing on patient biopsies that we had target engagement. We saw the shutdown of the mixed signature. And we saw this with dose response. So dose level 5 had a better shutdown than dose level 3. So this was very, very encouraging. And we also saw that the patients that had benefited by stable disease had much more profound mixed shutdown compared to the ones that progressed. Then we have also identified some interesting liquid biomarkers. So this is a pharmacodynamic biomarker, 3 different circulating factors that increase only in patients that benefit from our drug, never in the patients that progress. And this signature disappears once the patients start progressing. So this is really something that in the blood can give us a readout of whether our drug is being effective or not. We also have identified a predictive cytokine signature that we are now validating in the ongoing clinical trials. This has been elaborated by an artificial intelligence company called ABSU that has created 3 different predictive models that could help us stratify the patients that have more likelihood to respond to our drug. What is going on now? Just concluding. We have a phase 1b in first-line metastatic PDAC in combination with gem, nab-paclitaxel, and we heard something about it earlier. The good news is that we completed the recruitment. We have now all patients included in the trial, and the results will come out soon. We also have, just to mention it, a phase 2 ongoing in advanced osteosarcoma patients, so also pediatric disease. And we are also very excited about another trial that is starting in the United States, a window of opportunity trial at OHSU. Window of opportunity trial. These patients basically get diagnosed with cancer, and just before receiving surgery, so during the weeks in which they are waiting to receive the surgery, they can get an experimental drug. So they can get Omomic. That means that instead of getting just a biopsy, we get the tumor samples from the surgery, big chunk. The dream for biomarker studies. So this will complement very nicely our phase 1b, in which there is the confounding chemotherapy. In the window of opportunity trial, there is no confounding chemotherapy, and so the 2 things will be interpreted together. So this is just to tell you what we are doing in pancreatic cancer. I really hope to be able to give you news soon. The goal all along has been to provide patients with a new therapeutic option, safe. I want to stress that the safety of this drug is amazing. We have only grade 1, mainly grade 1 side effects that are chills and shiver at the moment of infusion. So this is really a drug that can be combined with a lot of other drugs. So with this, thank you for your attention, and I want to thank the people in my lab and at Peptomic that work really side by side, and when we talk about synergy, we usually think of them. So thank you very much.
Thanks very much, Laura. Great talk. Any questions from the audience? Yeah.
Hi, great talk and wonderful research. My question is, since your markers are cytokines and immune factors, have you characterized more of the immune system or what the role can be of the immune system in your system?
Yes, absolutely. It's an excellent question. We are doing a lot of studies. What we have seen is that the treatment with Omomic induces recruitment of T cells with a Th1, Th17 phenotype. And so we are characterizing now the activity of the different immune cells in this and their contribution. In mouse models, if we do the same treatment in immune deficient mouse, and then we humanize the same mouse with, basically with PBMCs, and we compare the same treatment side by side, every time we have the immune system there, we have a much more profound shrinkage of the tumors. So definitely the immune system helps us in the anti-tumor activity.
Here.
Yeah, really nice presentation. I'm curious about your PDAC patient, obviously. What was unique with that patient? Was this someone that did not have liver mets, or was a patient with liver and peritoneal disease? We do see ctDNA responses often and not always correlate with radiological responses, but obviously your patient is a great case because of the prolonged stable disease. Just curious about what was unique about this patient.
So you're absolutely right. Now, this patient didn't show, our N is so small in the phase 1 for the different oncological indications that it's really difficult to say what is so special regarding that one, right? So that's where we hope that a more homogeneous population, like the one in the phase 1b, will tell us more. This patient had liver metastasis, and also those had the notable shrinkage. Sorry, the other thing was... no, the ctDNA, sorry. Also in the phase 1b, the ctDNA correlates very nicely with the clinical benefit at the moment.
This was a patient who had already progression on a prior line of chemotherapy, yes. Right?
4th line.
Silvestre.
Yeah.
No?
Okay.
Yeah. So congratulations, I think great work as always. One question related to the, remodeling of the potential remodeling of the stroma. Have you had the chance to have like pre-post biopsies of your, Omomic treated patients to see whether there is any impact beyond the immune system? Maybe like, the fibrotic content changes,
yeah.
Any
so,
data there?
In the phase 1, we were a little bit limited in this type of analysis because we did the biopsy only 15 days after starting the treatment, so we didn't expect many changes. For example, the cytokine, the liquid biopsy signature appears very clearly after at least 4 weeks. So we don't expect these changes to be so fast. But we saw changes in the transcriptome. So we see definitely immune reprogramming already after 2 weeks. So it's happening, but at the histology level, we don't see it as significant.
Okay, last one, I'll do that. Have you considered to use that as a diagnostic tool?
Absolutely. In the lung study, it would be a very interesting use of it. Of course, we are not expert in that, and we would be happy to collaborate with somebody that develops diagnostic. So, yeah.
Thanks very much, Laura. Next speaker is Silvio Vicente. His talk is combination strategies to potentiate the effect of KRAS inhibitors. He is the group leader of Oncogenes and Effector Targets group from CIMA University of Navarra in Spain.
Okay, well, first of all, you hear me? Yes?
I would like to thank the organizers, Mariano and Carmen, for the kind invitation. Coming here to speak about anything related to KRAS is kind of like really humbling because, there's no authority such as Mariano who could be, like the one leading this field. Okay, so again, just as an introduction, in my lab, we have a long-standing commitment to study KRAS-driven tumors. We initially studied with lung. We now moved to pancreatic cancers and some other biliary cancers. The reason behind this is, as Laura was mentioning, referring to MYC, KRAS is indeed the most frequently mutated oncogene. And again, it's kind of like the big driver in among the oncogenes community. And what's important to the audience here is that in particular in pancreatic cancer, there are the frequency of the mutations is over 90% in the patients. So this means that despite we know from some studies that we can find KRAS mutations even in wild-type tumors, and in the context of oncogenesis and the fully formed pancreatic cancer tumors, KRAS stands as a very prominent target. So if we have a look at the history of how RAS proteins were initially identified and the transition towards the development of new or potential therapies to treat these tumors, we've seen many examples of failure over the years, starting with the farnesyl transferases, then the RAF/MEK inhibitors, then this wave of synthetic lethalities, which was quite disappointing at the time. But now, from about 10 years far from now, the group of Kevan Shohats, basically, they instigated a new kind of like commitment in the field because they were able to develop the new direct or the first direct inhibitors to KRAS. After that, there were some attempts to optimize the molecules until sotorasib and adagrasib were approved for the advanced non-small cell lung cancer, and combinations of those with EGFR inhibitors are also approved now for colorectal cancer. However, if we have a look at the survival curves, we can see, and these are the curves for lung cancer on top and pancreatic cancer on the bottom, for the approved drugs, they are far from being curative mutant KRAS tumors. And this is an issue that we have to face, and now we need, as a community, we need to come up with new strategies to come up with combinations that could be more effective to those anti-tumor effects observed as single agents. So in the lab, basically we have taken we have adapted to how the field was moving in the BC area, what we consider before the chemical inhibitors to KRAS era. We were trying to understand more profoundly the RAS-regulated network in an attempt to find new potential molecular targets that we could use as potential therapeutic targets. Now, with the availability of some RAS inhibitors and the current status in the lab where we have access through some agreements with companies to investigate those new inhibitors, it's also important to come up with a setting where we can integrate the understanding the molecular networks to be more efficient in designing new combination strategies. And in the lab, we have basically taken a very integrative approach using different type of models shown here. Also, translational studies incorporating human biopsies, PDX samples. We have made a strong effort on new computational analysis, especially drug repurposing analysis, and we're working closely with the computational groups in THEMA. And now, although we are kind of behind the field, we are now trying to introduce single cell and spatial technologies, which we hope are going to enable us to resolve some aspects that we haven't taken into account as up to now. So if you have a look at this slide here, the initial approach that we took in the lab, this was like over 10 years ago when I started the lab, it was very simplistic. So we basically were aware of the multiple changes that were ignited by oncogenic KRAS, and this could be also applied to KRAS inhibition these days. However, we basically went on with a very simplistic hypothesis, and this was that maybe within the KRAS distal output, we could find KRAS effectors or gene signatures that could open the possibility to novel therapeutic strategies. And this is basically work that we consider to be in the busy area, but today we'll try to give you also an example of how we have transitioned also to working with the new RAS inhibitors. So the premises that we took in the first place to try to understand the KRAS distal output were the following. So the first one, we were trying to focus on those changes that occur early in the evolution of the tumor and basically were sustained across the whole the carcinogenesis process. And this is kind of like mimicking this idea of tumor evolution through genomics, where normally we have these truncal mutations, which are normally considered to be the more robust therapeutic target. So we were kind of like focusing on at the gene expression levels based on this concept. The next concept that we wanted to integrate was to try to go after those KRAS-regulated elements that were expressed across different KRAS-driven tumors, assuming that eventually we could use those as a way to treat or to reach larger fraction of patients in the case we could come up with new therapeutic strategies. As Paco was mentioning earlier, and also very nicely illustrated by Merichel during her talk, we also tried to have interspecies analysis because we wanted to go also after those conserved molecular mechanisms which we believe are more representative of what is the KRAS biology, and in that sense maybe being more central to potential targeting. And then obviously we prioritize all the genes or gene signatures based on their clinical relevance or potential translatability based on available drugs. So the first question that we came up with or one of the first questions that we had in the lab was, can we uncover a core gene expression signature which is representative of mutant KRAS cancers? And this work that basically represented the foundation of multiple studies in the lab, which started by basically analyzing gene expression data from different tumor types, which basically had in common the expression of oncogenic KRAS. So we derived this experimentally derived signature, and then we filter it through a series of patients, over 1,000 patients, comparing mutant versus wild type. And then we basically focus on those genes that were recurrently present in the leading edge of genes expressed in the mutant KRAS patients. And as a result, we came up with a 8-gene signature, which we call the cross-tumor KRAS-on signature, because it was representative of the KRAS-on state. These days, if you have been following the field, the group of Channing there, they have described now a kind of like KRAS-off signature, which was published in Science in a few months back. So basically, the first thing that we decided to do was to study this signature at the clinical level, so basically to see whether it had any translational value. And what we could observe is that those patients that had high expression levels of the signature, along with mutations in KRAS, displayed the worst survival. And this was also true for cholangiocarcinoma here. And again, in the case of pancreatic cancer, where we could not do that stratification due to the high frequency of the mutations in patients. So this was nice, but again, it was not functionally relevant. But then, over the years, we have moved to dissect different elements of the signature, particularly FOSL1, which we describe as a vulnerability both in lung and pancreatic cancer. Similarly, PITPNC1, also in lung and pancreatic cancer. And a similar approach was undertaken to study microRNAs, and we could also identify the MIR181AB1 cluster as a functional microRNA in both lung and pancreatic cancer. And indeed, in some examples, for instance, the FOSL1, we've been able to transfer the knowledge to other additional KRAS mutant tumors, which is cholangiocarcinoma. So again, we had shown so far that the KRAS-on signature is clinically relevant, and it also includes functionally relevant elements. So with this in mind, we decided to investigate whether we could use this signature to uncover potential drug combination strategies. And this work that was done or has been done by Sruthi here is a PhD student who has just defended her thesis, and Oyane, who was a postdoc, is now in another lab. So going back to the 8-gene signature, we observed that there were a couple of genes which were part of the laminin 332. It is a component of the extracellular matrix, which has been involved in the induction or in the context of oncogenesis in processes related to metastasis, proliferation of the cells, and in some cases also to respond to specific treatments such as chemotherapy. And one of the ideas behind focusing on this, or basically because these two genes were laminin, we decided to focus on pancreatic cancer. Because in pancreatic cancer, I mean, as we all know, it has a highly desmoplastic stroma, where one of the components, non-cellular components, are the laminins. So we are very aware that we have obviously stromal cells, either normal fibroblasts or CAFs, the non-cellular collagen component, which is important. This normally leads to an immunosuppressive environment, but again, laminins, because they are also kind of like, they play an important role in pancreatic cancer. In this case, we focus on this particular tumor type. So in order to see whether, again, this expression of the laminins and other components of the KRAS signature, we performed a meta-analysis, so we could basically integrate all the RNA-seq and microarray studies that we can find on pancreatic cancer comparing normal and pancreatic tumors. And basically what we could see is that among the top scoring genes, we had LAMB3 and LAMC2. So we didn't focus on LAMB3 because at the time that we were kind of like interpreting this data, the group of Jorge Moscat, when he was in San Diego, they basically showed in collaboration with another group that LAMB3 was a synthetic lethality in lung cancer. So we decided to focus on LAMC2. So because the data that we had used for the meta-analysis was not taking into consideration the individual cells, so we decided to move and had a look at this study here that Merichel has explained before. So these are 24 tumor PDAC samples and 11 normal samples. And what we could see, which was quite interesting because we didn't anticipate this, basically most the tumor, I mean the cell type that was expressing LAMC2 at the highest levels were the kind of like the transformed ductal cells or neoplastic cells. So this was confirmed again in a series of, in a TME of pancreatic cancer patients in collaboration with Vincenzo Corbo. So we could see, just as a global view, that around 75% of the patients express relatively high levels of LAMC2. And we could see that the expression levels were found mainly in the tumor cells, as you can see here, with a different range of expression of LAMC2. We didn't find any expression in the Langerhans islets or even in normal pancreas ducts. And this was kind of like also mimicked in cellular, in cell lines. Around 70% of the cells had high expression or the detectable expression of LAMC2. And also the KPC model and multiple cell lines derived from this KRAS-driven mouse model also had high expression of LAMC2. One interesting aspect is that we could start detecting LAMC2 even at the banning stage, suggesting this could be kind of like a perineoplastic biomarker. So moving on to functionally validate LAMC2, we first established cultures in 2D and 3D. And one thing that caught our attention is that a high expression of LAMC2, high expression of LAMC2 here basically identified those cells that were more proficient in forming 3D structures. And this kind of like related to work from Elsa Lombardo's lab, where they've seen that LAMC2 fosters the cancer stem cell phenotype. And if you remember, normally those conditions that are applied to generate these 3D conditions favor the stem cell phenotype. So that was nice. And then we basically showed that inhibiting LAMC2 basically impaired the formation and the initiation capacity of the 3D organoids. And basically, as a consequence, when we did the LAMC2 inhibition in already established tumors, we could see that it also basically delayed the tumor growth. So up to here, we had kind of like functionally validated the the role of LAMC2. So we wanted to study a bit more what was the network regulated by LAMC2 to this with RNA-seq. And we could find that the gene signature that was downregulated by LAMC2, basically those genes that we could infer that are more oncogenic, were highly enriched, as you can see here, in pancreatic cancer tumors. And also we could see that these signatures were predictive of poor survival in 2 independent data sets. We moved to analyze by geo the categories that were enriching this signature, and in the first place we could see many categories related to growth factor and receptor tyrosine kinase interaction. So one of the top hits here was AXL, the receptor tyrosine kinase AXL, and we decided to incorporate it into the workflow because AXL had been already described to be a potential target in pancreatic cancer, sensitized to chemotherapy, and also might be involved in plasticity in this type of tumor. On a second level, we also found that the FRA pathway was upregulated, so we had already reported that FOSL1, which is a member of the FRA pathway, it was important in pancreatic cancer, so we decided also to focus on this part. And indeed, one thing that we found is that using FOSL1 signatures that we had generated in the lab in different tumors, all of the signatures were downregulated in the context of the LAMC2. So everything was looking fine. So we decided to test whether LAMC2 was indeed regulating AXL, and this is what we see here in the upper part in cell lines, and then here's in the bottom part in xenografts. Something similar was done for FOSL1. And then we checked with the promoter scanning for AXL. We saw some AP1 binding sites, and what we found is that there was a nice correlation in the 2 data sets that we had investigated, and indeed when we depleted FOSL1, AXL levels went down. So we went to construct a very simple model. So we have LAMC2 regulating FOSL1 and AXL. We had not kind of like formally tested whether AXL could be regulating FOSL1. It's a plausible explanation, and this I will explain in a few slides from now. But basically, we took an approach to combine these 2 drugs that one is specific to AXL and then MEK, which could be regulating FOSL1, to kind of like go after maximal inhibition of the pathway. So by doing this, we could observe that the combination was more effective in affecting the cellular viability, and this was a setup that we have no longer adopted because these are treatments for 72 hours. Normally now we go with for either 7 days or 10 days, where we normally have better results. But in any case, again, the combination was working better, and then this was, this was working through an induction of apoptosis. We went on to test this in mouse-derived organoids, so basically organoids derived from primary pancreatic cancers in the KPC model, and we could see that again, both the organoid size and the number of organoids were compromised in the context of the combination. And in collaboration again with Vincenzo Corbo, we tested this also in patient-derived organoids to kind of see the same result. So up to this point, for us, it was surprising to find out that Channing was having similar results using an ERG inhibitor. So he ran a drug screen to look for sensitizers to ERG inhibitors. And what he saw is that one of the top hits was an AXL inhibitor. And basically, this is a summary of the results. But at the time, we met at a meeting, and we said, well, maybe we can collaborate on this project somehow. Because the obvious question that arises from this data is, what if we replace MEK inhibitors or ERG inhibitors by a RAS inhibitor? And from those who have again followed the RAS field in the last years, there has been an explosion of RAS inhibitors going beyond those targeting the G12C mutations, and now we have some others that target KRAS G12D, G12V, some wild type because they go to KRAS amplifications. but some others which can be considered pan-RAS inhibitors. And we focus, or we took advantage of this inhibitor because both Channing's lab and my lab had access at the time to the inhibitor, and also because there was compelling data in these back-to-back studies in Nature suggesting that this would be a good candidate to look after in the field of KRAS. So Channing, from one side, they started to run some CRISPR screens, and what they saw is that some of the genes that were sensitizing to this inhibitor were AXL. So basically, this was consistent with our hypothesis that AXL could combine well with inhibitors against the RAS pathway. And to kind of like try to interpret and to come up with like some new studies or analysis to understand the mechanisms, basically it's important to understand how AXL functions. And AXL functions obviously by is binding to this ligand Gas6, which is going to initiate the whole signaling cascade. It can control the RAS pathway, basically can act as a reactivator or to sustain the pathway in the context of inhibitor to the RAS pathway, but also basically control some other pathways that have been described, for instance, PI3 kinase or JAK kinase, and this is a study that we published a couple of years ago, which can act as a compensatory mechanisms that also basically whose abrogation could also sensitize more deeply to the RAS inhibitors. So the first thing that Chinese group did was to check for overexpression of AXL or the ligand, and where there some differences in the AXL levels depending on the cell lines and also the concentration of the inhibitor, upregulation of Gas6 was more consistent. So we are now also looking at the activation levels of AXL, which might be a better readout than just AXL expression. And this was also found at the mRNA level, as you can see on the right-hand side. So in my lab, we went on to test this inhibitor in multiple cell lines. I'm here giving you some examples of human and cell lines. And basically, what we could see, again, using these matrices, that the Bliss score for the combination of the AXL inhibitor and the RAS inhibitor were very compelling. And this data not only was found in 2Ds, but also in 3D PDX cultures. Okay, so again, there was a consistent synergistic effect of the combination across the different cellular systems. So we then went on to start doing some in vivo experiments, and we basically took the lead in my lab in doing those. And basically, as you will see here, we only have done treatments for 15 days. So obviously, these are conditions which would be like minimum and not obviously representative of what you would do in the clinic, but even under those conditions, we have seen in cells that are intrinsically sensitive that again the combination works very well, given to tumor regressions, that these are at suboptimal doses because these cells are very sensitive in the first place. And what's interesting is that we have the survival or the time to 1,000 millimeters cubic was extended in the context of the combination. This was true also for another cell line, which is the KP4, which is kind of like a recurrent cell line, which is, or a cell line which is recurrently resistant to all the RAS inhibitors that have been tested to date. And even in these conditions, again, on the RAS inhibitor only arm, we can see that the cells of the tumors grow well. However, in the combination, we have a strong synergistic effect. And again, the survival is extended. So as a proof of concept, we moved to run these experiments, to run some RNA-seq experiments on the tumors that were treated with the different arms, so the single AXL RAS inhibitor and the combination. And one thing that was expected, or at least from our hypothesis that we were, with the two drugs, we're going to have maximal inhibition, was basically the enrichment or the negative enrichment of the KRAS ERK signaling pathway. But then we also saw some enrichment or negative enrichment of signatures that had been related to potential mechanism of resistance. And one was MYC, but MYC, because of the time point at which we did the experiment, could be just a consequence also of the RAS inhibition. However, we also saw that the YAP pathway was also depleted, and YAP has been shown very nicely in the last years by several groups that is a mechanism of resistance to KRAS inhibitors. So again, the combination not only is maximally inhibiting the pathway, but also maybe abrogating potential resistant mechanisms. To confirm this, basically we moved to test initially for the RAS activation. As you can see here, we have a clear inactivation of the RAS, which is more deeply than the single treatments. And this was accompanied, depending on the model, with a decrease in the proliferation and also increase in the apoptosis. And in the case of the more resistant model, again, for the ERK inactivation, the trend was similar. And here we only observe increased apoptosis, as you can see here in this graph. So these had been, the experiments so far had been done in immune deficient mice with human cell lines. We then moved to immune competent mice. This is, again, the first example, which was done just subcutaneously to see whether we could see, which were the results that we could observe. And as you can see here, again, only with the 15-day treatment, we can see a lot of regressions in the context of these subcutaneous tumors. But because in the context of pancreatic cancer, again, this was also accompanied by an increase in survival. But again, these models are not fully representative of what we would expect in the therapy field. Normally, we, as you know, Mariano, well, in this Vasiliki and also Dieter have shown, the community is now moving preferentially to orthotopic models, so we also did this. And what we can see here, we started treating tumors with the combination that in this experiment were around 100 millimeters cube, and you can see that we can have a tumor control or tumor regression quite compelling in the combination. And this was, again, accompanied by a decrease in ERK activation and an increase in apoptosis. We repeated, or we launched a different experiment to assess survival. In this case, for logistical reasons, the average of the tumors were 150, but even under those circumstances, we could also see that there was an extension in the survival of these mice. So the data altogether suggest, especially from the cellular models, because, the human cell lines to mouse cell lines comparison, that the effect is mainly cell autonomous. But we want also to test whether there was any influence on the tumor microenvironment of the tumors in the context of the combination. This is a summary of some of the populations. We have assessed like 8 to 10 populations in the lineage, in the lymphoid lineage, and then 8 or 10 also in the myeloid lineage, but here just are just the most representative. We didn't see much changes for CD8, CD4, or NKs, and also for dendritic cells, but we could see a change in the ratio of M1 to M2 macrophages. And this, we investigated a bit more in depth, and what we can see is that in the context of the combination, the M2 population is lost, and there is also a decrease in the fraction of macrophages that are transitioning towards M2 phenotype. So it seems that in addition to cell autonomous effects, there might be some influence on the tumor microenvironment. And with this, I'm just going to close. So basically, I've shared with you that the LAMP2 is upregulated in human and mouse pancreatic cancer. It might represent a molecular target. LAMP2 regulated network includes targetable effectors such as AXL and also central transcription factors. So FOSL1 has been not just described by our group, but also Skoldo's group and some other groups basically is involved quite strongly in the plasticity of the cells. That combined AXL and MEG inhibition could be a efficient strategy, but for sure now in the context of the RAS inhibitors, it might be more ideal to use this inhibitor. And again, there might be cell autonomous and non-cell autonomous effects in this phenotype. With this, I'm just going to close. Thanking all the people involved, all the collaborators, Vincenzo Corbo, Jens Siveke, Chaninder, Christine Sers, Perfect Secretary at Stanford, and also the people from Revolution Medicines who have been the contact to and provided the inhibitors, and also the funding sources. And with this, I will be happy to take any questions.
Thanks very much, Silvestre. Any question from the audience? Yeah.
So this was fantastic. Are you planning to move this to the clinic? Have you identified a AXL inhibitor that you would like to work with? Because I know a company with an AXL inhibitor and they will love to develop this in pancreas.
So I'm not sure if it's the same company that Channing is now, like pursuing, because this is something that I think is being attempted for the last 2-3 months. We are now trying to involve Revolution Medicines for sure. But again, if the company is not the same that, as that Channing is, working with, I think we need to talk, because again, one thing that, it was unexpected to us is that, at least from working in lung cancer by Eric Haura, we were talking about this before, AXL seemed to be just a mechanism of resistance for specific subtypes. For instance, in the mesenchymal, those tumors which are more mesenchymal. But here, at least, from the models that we have tested, it seems that it's a widespread mechanism, right? So, again, there are changes, like tumor-to-tumor changes, phenotypic changes, which would influence that. But I think it would be a very strong target for a combination.
Okay, if there is no other question.
Thank you.
Thanks very much. We're going for our next speaker, that is Teresa Macarulla. She is presently the head responsible for the Department of Medical Oncology at the Clinic Hospital in Barcelona. And she's going to talk to us about the state-of-the-art and new options in advanced pancreatic cancer. With pleasure.
Thank you very much for the presentation. So thank you, first of all, for the invitation to be here today, and congratulations for this, I think, very, very interesting meeting. So now we will change a little bit to more clinical data, and I will present in the next 15-20 minutes the new potential treatment options that I think maybe can change the treatment in patients with pancreatic cancer. So it was mentioned before, but I think it's so important to repeat again that pancreatic cancer is a cancer with worse 5 years overall survival. So really we had an urgent need for the new treatment options in order to improve the median overall survival of our patients. How we treat pancreatic cancer today, the advanced pancreatic cancer today. So unfortunately, in this slide, you can see the treatment options that we have today. That is only chemotherapy. In fact, here I put nalirifox, that is liposomal irinotecan plus 5-FU oxaliplatin, but this is only approved in some countries. For example, in Spain, it's not approved, so this is not a possibility for us. Gemna-paclitaxel and FOLFIRINOX. And with this is the treatment option for our patients, first line, second line. We combine both strategies. The median overall survival of our patients, as you well know, is less than 1 year. So really we have, I think, to improve these really poor numbers. So this is my outline, how I think I put here, except KRAS inhibitors, that of course, I will not review this part. I think it's the most promising part. In fact, I think this is, here is my hope, but even with this, Ignacio will review it very well after me. So I will review the other part, the targeted therapies in pancreatic cancer, the still some immune strategies, the HRD, and finally the ADCs as a potential treatment option for our patients. Starting with targeted therapies in pancreatic cancer, as you well know, in our group, published, led by Florian Castet, the characterization of early onset pancreatic cancer. You know, this is a very important population for us because it's increasing, and that means patients diagnosed with 50 years or less. And we describe how in this population, the KRAS wild-type proportion of patients is higher than patients with average age, and also we demonstrated that this population presented higher incidence of potential targetable alterations. Other groups, for example, this publication in the United States demonstrate the same, but also they describe up to close to 40% of patients with KRAS wild-type tumor presented potential targetable alteration. That means these fusions, these BRAF mutations, these genes amplification, and that's important to take into consideration. This is some examples that we have of targets of KRAS wild-type tumors. For example, NRG1 fusion, that is almost exclusively of patients with KRAS wild-type tumors and young patients. That means younger than 60 years old. And we have this data, very nice data of cenacotuzumab, that is a bispecific antibody against two specific locus of HER2 and HER3, and we have these 15 patients responding out to 36 patients that were treated with this drug in a refractory setting. That means patients previously treated with 1, 2, or 3 lines of chemotherapy, and they presented 42% of patients that presented a partial response. That means the tumor, the reduction of the tumor was higher than 30%. So that means that's better than what we have with chemotherapy in first line. And then, and importantly, the duration of this response were really high, 9.1 months, that again, this is superior of the median overall survival that we have, for example, with gemcitabine. So this is a very good strategy, but with this small number of patients with this specific fusion. Then we have another example, for example, patients with RET fusion. Again, this is exclusively of those patients with KRAS wild-type tumors, and in the Libretto trial, that it was a trial that included patients with solid tumor and RET fusions, they include 12 patients with pancreatic cancer and RET fusion, and they demonstrated this response rate of 54%. So half of the patients respond to this therapy, and the duration of response was not reached at the moment of the publication. So really those patients were treated with selpercatinib, that is an inhibitor of the RET protein. So that's, again, a good strategy for this small number of patients. Let's not change. Now it's not a KRAS wild type. Now we talk about another target, that is Claudin 18.2. And I will start with the drug that is more advanced in the development of a drug against Claudin 18.2, that is zolbetuximab. Zolbetuximab is an antibody that is directed to this Claudin 18.2, and it was tested in a randomized phase 2 trial, include patients with pancreatic cancer that presented a high expression for immunohistochemistry of Claudin 18.2, and that was described as the patients that presented more than 75 of tumor cell with moderate to strong Claudin 18.2 expression. And those patients were randomized to be treated with only chemotherapy, gemcitabine or gemcitabine and zolbetuximab. We are waiting the results of this important trial, and probably it will be presented in the next ESMO meeting in 1 month. We have another drugs that test also that targets Claudin 18.2, and this is an antibody drug conjugate that specifically targets Claudin 18.2. And this drug, BI3.3, is a drug that demonstrated an important activity in patients with, again, high expression of Claudin 18.2. In this case, they define high expression differently than zolbetuximab. They describe by immunohistochemistry an expression of 1+, 2+, or 3+, more than 60% of tumor cells. So as you can see, every drug has different cutoff of the positivity of Claudin 18.2. And here they demonstrated in a really refractory population, 26% of the patients were previously treated with more than 3 lines of chemotherapy, so really this is refractory. They demonstrated the 20% of response rate in patients with high expression Claudin 18.2, because they demonstrated no activity in patients with no expression of Claudin 18.2. So you need some expression of the protein. So this is really promising, and then they also demonstrated this PFS of 5.4 months. That is the same PFS that is described for gemcitabine in first line, and this is in third or fourth line, so really promising, and a median overall survival of 9 months. Let's see how this drug moves forward, but really this data presented at the last ASCO meeting was really promising. And then we have another mechanism of action, that is this IB389 drug, that is an anti-Claudin 18.2 and CD3 bispecific antibody. So the drug, the antibody binds to these two targets, and they demonstrated, again, in a positive pancreatic cancer, but they define positive like immunohistochemistry 2 or 3+ in more than 10% of the cells, so again, different cutoff, they describe this overall response rate close to 30%. So again, this is activity in a refractory population and seems promising in this specific group of patients with different but some expression of Claudin 18.2. And I also want to highlight this drug that is an ADC against Claudin 18.2. It's the drug of AstraZeneca. I want to highlight this because we don't have data of the phase 1 clinical trial with pancreatic cancer, but we have this trial that is ongoing, that is this ADC directed to this Claudin 18.2 specific drug in combination with chemotherapy. So the ADC contains chemotherapy, this cytotoxic microtubule agent inside. So that means that we have to be careful when we want to combine these drugs with chemotherapy. And in this case, in first-line pancreatic cancer, they put together this drug plus 5-FU and irinotecan, like a FOLFIRINOX, but with this ADC, or this drug with gemcitabine. So let's see what happened with the toxicity and how we can manage these combinations, but this is ongoing. Probably we will have data next year, and the question is how we will move forward in first line or in second line with these combinations, because we have to think that these one day maybe have to compete with the data that Ignacio will explain us with the KRAS inhibitors, so we have to think how to move forward with these strategies. So let's now change the target again, independently of the KRAS mutations, that is the MTAP deletions. So deletion, we will review different drugs just in order to see that this is really an active field in pancreatic cancer. We have this drug that is the Amgen compound, the AMG 193. This is a PRMT5 inhibitor that was tested in this phase 1 clinical trial with different tumor types that presented or CDKN2A deletion or MTAP loss. And they tested different tumor types, but they included 23 patients with pancreatic cancer, and they demonstrated that 5 patients presented partial response, so some activity. Interestingly, they demonstrated only activity in patients with MTAP loss. And now they are running a phase 1 clinical trial that combines this drug plus chemotherapy, gemcitabine or FOLFIRINOX. Interestingly, because this drug caused some GI toxicity, so we have an overlapping toxicity with these chemotherapies, and interestingly, the combination with FOLFIRINOX presented better tolerability than combination with gemcitabine. And also interestingly, they will also try this drug in combination with the pan-RAS inhibitor of Revolution Medicine in second line. So there's a synergistic effect between these two strategies. And then we have another drug that is a BMS, a PRMT5 inhibitor, that again, in a phase 1 clinical trial with different tumor types, but with MTAP loss, they tested the drug with 35 pancreatic cancer patients, and they demonstrated 17% of patients with partial response. And now they are planning a randomized phase 2/3 clinical trial in patients with first-line metastatic pancreatic cancer and MTAP loss, and those patients are randomized to be treated with gemcitabine or gemcitabine and the PRMT5 inhibitor. And finally, we have this drug that also is interesting, is from the Tango company, and they is a PRMT5 inhibitor. They test this drug in a phase 1 clinical trial, again with patients with MTAP loss, and they still don't presented data from this phase 1. They presented and published data, preclinical data in pancreatic cancer in mouse models showing this activity in patients in models with MTAP loss, but we still don't have the data of the monotherapy, the phase 1/2 monotherapy trial, but they are planning a phase 2/3 clinical trial, again, in this case in second line with this drug in monotherapy in patients with pancreatic cancer and MTAP loss, or the patients are randomized to this strategy or the standard therapy that is chemotherapy. So let's see the results that will be presented next year. And also it's interesting that again there's a phase 1/2 clinical trial ongoing in the United States in which they are testing this drug in combination with RAS inhibitors or G12D inhibitor or pan-KRAS inhibitor in patients with MTAP loss and KRAS G12D mutation. So this is an interesting target. There's a lot of activity here, so probably in the next years we will have the results of these phase 3 clinical trials, phase phase 2, 3 clinical trials with randomized data, and maybe here we have an opportunity with this 20-25% of patients with pancreatic cancer and MTAP loss. And I want only to mention this drug because this is a pathway that in general we are not treating in pancreatic cancer. This is a randomized phase 2 clinical trial that was presented this year at ASCO. It's this drug that is elraglusib. This is a novel cell GSK3 beta inhibitor. We know that this GSK3 beta pathway are promoting the cell survival, the proliferation, suppress apoptosis, are implicated in the chemotherapy resistance. So it makes a lot of sense to block this pathway, and they demonstrated in a randomized phase 2 trial that when they combine this drug with chemotherapy, compared with chemotherapy alone, this had an impact in overall survival, as you can see here, 10.1 months compared with 7 months. So it's really something that it seems active. Then they didn't demonstrate an impact in progression-free survival, so patients progress equally, and slightly higher response rate in patients treated with experimental arms. So let's see what happened with this drug. Interestingly, they presented data from the translational research of the trial, and they demonstrated that there were some elevated cytokines correlated with better survival, and some decreased cytokines correlated with increased survival. So interestingly to see maybe something that can guide or can help to move forward with the development of this drug. Now we will change to immune strategies in pancreatic cancer. As you know, we fail in all the clinical trials in which we try different strategies, different immune strategies to treat pancreatic cancer, but we still have some clinical trials ongoing with the hope that can move forward. This is one of the trials. This is a CD40 agonist, mitazalimab, that in a phase 1-2 clinical trials with around 60 patients in first-line pancreatic cancer treat the strategy of this CD40 agonist plus FOLFIRINOX. And they started with the CD40 agonist, in theory, to change the microenvironment, and then they add the FOLFIRINOX, and then after FOLFIRINOX, we give the patients the CD40 agonist. Interestingly, they demonstrated the response rate of 42%. This is higher than expected with FOLFIRINOX alone, that is around 30-35% of response rate. And again, the median duration of response, when the patient responds longer than we expected with FOLFIRINOX alone, is 1 year. And with FOLFIRINOX alone, the median duration of response is around 6 months. So it seems it can be a little bit more active. This activity is enough to move to randomized trial, phase 3 randomized trial? We are not so sure. Let's see. Again, interestingly, they describe an association between this CD40 cells expansion with the better overall survival. However, we have another CD40 agonist, that is this sotigalimab, that demonstrated negative results in combination with chemotherapy. That means that we have a previous trial that was negative. In this phase 2 clinical trial, patients in first-line metastatic pancreatic cancer were randomized to be treated with this CD40 agonist plus chemotherapy, or with nivolumab, a PD-1 inhibitor, plus chemotherapy, or the combination of CD4 agonist plus immuno plus chemo. And interestingly, they demonstrated that the best response were achieved, or the best overall survival were achieved, those patients treated with nivolumab and chemotherapy. And that's interesting because we have a negative trial, a phase 3 clinical trial with a combination of nivolumab and chemotherapy. So really, the combination of CD40 agonists was not or failed in this clinical trial. And however, they did a huge effort to try to find biomarkers in order to see which are the patients that can respond to this strategy, and they define a biomarker or survival, predictive biomarker related with the survival after the arm of nivo chemotherapy, and also for the CD40 agonist and chemotherapy. However, if this is enough to move forward only for this population or not, it's probably not clear, and probably it's not enough. And then we have another strategy that this is the phase 1 in which we treat the patients with pancreatic cancer with a combination of this oncolytic adenovirus plus chemotherapy, gem/nab-paclitaxel. This was the phase 1, and you can see here that the response rate, half of the patient responds to the therapy. Of course, it was the chemotherapy there, but the response rate was higher than expected only with chemotherapy because it was half of the patients. And interestingly, after these results, we ran a phase 2 clinical trial, randomized. So patients were randomized to be treated with the oncolytic adenovirus plus gem/nab-paclitaxel or gem/nab-paclitaxel alone. And the results are really not bad. Will be presented in the next ESMO meeting in 1 month in Berlin. Interestingly, in the phase 1 clinical trial, we had some patients with baseline biopsies and also biopsies at day 8. That means the patients that the oncolytic adenovirus was administered at day 1, and then the chemotherapy started at day 8. So the biopsy was before the chemotherapy, and they can demonstrate this induced inflammatory response in the liver, and this is the data on the liver in the patients treated with the virus, and it is very interesting. So also, I want to highlight this trial mainly because it's a phase 3 clinical trial. That means that if the trial is positive, can change the standard of care in pancreatic cancer. That this is this drug that is the quemliclustat. This drug is a CD73 inhibitor. And in a phase 2 clinical trial, they demonstrated promising activity of this drug in combination with chemotherapy. When they tried to add a checkpoint inhibitor plus this CD73 inhibitor and chemotherapy, they failed. There was no synergistic effect. So they decided to run a phase 3 clinical trial in which patients with metastatic pancreatic cancer in first line were treated with chemotherapy alone, gem/nab-paclitaxel, or were treated with a combination of gem/nab-paclitaxel and this drug, quemliclustat. The enrollment is completed. We enroll really, really fast. That means that the necessity that we have of new treatment options, and we will wait the results of this trial that potentially can change the standard. And also I want to bring this because it's interesting to see TTFields. It's a device that in combination with gemcitabine and nab-paclitaxel in patients with locally advanced pancreatic cancer, that means patients that are not candidates for surgery but with no metastasis, these TTFields demonstrated an impact in the overall survival of the patient. So patients live longer if they treated with the device plus chemotherapy compared with chemotherapy alone. And interestingly, it has this device that it was located in the abdomen, it has an impact in the distant metastases. So there's some preclinical data, not in pancreatic, but in other tumor types that show how this device, these TTFields, can change the immune microenvironment of the tumor and also in the metastatic setting. And in fact, we ran a phase 2 clinical trial after this phase 3 only in locally advanced disease. Then we ran a clinical trial with metastatic pancreatic cancer, and patients were treated with chemotherapy, gem/nab-paclitaxel, plus immunotherapy, checkpoint inhibitor, atezolizumab, plus this device, TTFields, in order to see if the effect of this device can change the microenvironment of the tumor and the immunotherapy can be active in this group of patients. And we are waiting the results of this trial. And then we'll now change to the DDR, this specific group of patients with DDR alterations. You know that the core DDR alterations in pancreatic cancer are BRCA1, BRCA2, or PALB2 that are around 7–8% of patients, in Spain a little bit lower. And we know that this alteration can sensitize, give us the information that those tumors are more sensitive to platinum-based chemotherapy and also to PARP inhibitors. We have one phase 3 clinical trial that established the olaparib as a treatment option for this population, not in our country, that is not approved, but in the countries in which is approved. And in this trial, we screened like 3,000 patients with metastatic pancreatic cancer, and we found that approximately 7% of the patients presented this germline BRCA. Those patients were treated with platinum-based chemotherapy at minimum of 16 weeks, and those patients without progression can be randomized to be treated with olaparib, that is a PARP inhibitor, or placebo. And the trial was positive for its primary endpoint, that was progression-free survival, and really was superior in patients treated with olaparib compared with placebo. However, there was no impact in overall survival. Both group of patients presented the same overall survival, 19 months. But interestingly, the patients that were treated with olaparib and response to the therapy, that were 23% of the patients, the median duration of response was 2 years. So really 2 years, that's incredible for a pancreatic cancer patients. However, we have here in the left side of the screen that we have these 9% of the patients that were the long responders, patients that can live or can be treated with a drug for a long period of time. We have these 64% of the patients that presented this median overall survival of 18 months, what we can expect with this therapy. And then we have this group of patients, 25% of the patients, this is really high, that presented this median overall survival of 4 months, so patients that progress very rapidly to the PARP inhibition. That's interesting because today we do not know who are the patients in each of these boxes because all of them presented germline BRCA mutations, and all of them presented no progression to platinum-based chemotherapy. And today, this is moving to perioperative therapy. This is the Apollo trial, a phase 3 clinical trial that are testing this strategy, the olaparib, in patients with germline BRCA1, BRCA2, or PALB2, but in the adjuvant setting. Patients treated with surgery, and then with the standard chemotherapy, and then patients are treated with olaparib or placebo. So let's see if we can reproduce the benefit. And when we try to see beyond BRCA, because in Apollo trial we have only germline BRCA1 and BRCA2, we have this trial. This is a single-arm phase 2 trial that treats patients with germline BRCA1, BRCA2, PALB2, but also somatic BRCA. And we see that those patients, again, were treated with platinum-based chemotherapy, and if there was no progression, those patients were treated with rucaparib, that this is another PARP inhibitor. What it is interesting is that we can see response in patients with germline PALB2, so we can treat also those patients with PARP inhibition, and also in patients with somatic BRCA. Of course, again, when you treat somatic BRCA patients with PARP inhibition, some of them respond and some of them never respond to this strategy. And then we have the strategy to combine PARP inhibitors plus immunotherapy. And we have this very nice trial that in which patients with no selection, only 15% of the patients included in that trial presented DDR alterations. Patients that presented no progression to platinum-based chemotherapy were treated with niraparib, that is a PARP inhibitor, plus nivolumab, a PD-1 inhibitor, or niraparib plus ipilimumab, a CTLA-4 inhibitor. And interestingly, they demonstrated that there was more synergistic effect with PARP inhibitor and CTLA-4 inhibitor than with niraparib and nivolumab. As you can see here, the 6-month PFS was 20% with nivolumab and close to 60% with ipilimumab. So it seems it's important which drug and which target we are targeting. And we have also this interesting trial in which they try, this is the Polar trial presented last year at ESMO, in which we try to demonstrate if the PARP inhibition plus immunotherapy is a good strategy beyond germline BRCA patients. And they treat 3 cores of patients. One of them, HRD, core HRD, no problem, BRCA1, BRCA2, and PALB2. The second one, non-core HRD. ATM, CHEK1, other HRD, but not the core. And the other one, it was a cohort that they treat patients that respond very well to platinum-based chemotherapy, but not a specific mutation. And they treat those patients with pembrolizumab, immunotherapy plus olaparib, and they demonstrated only activity in those patients with a core DDR alteration. So only the patients with BRCA1, BRCA2, and PALB2 mutations. They demonstrated no activity or poor activity in patients with no alterations or in patients with other DDR alterations beyond BRCA and PALB2. This is the progression-free survival with the same message. And it's interesting because we, in the TTD, we completed this clinical trial with the combination of olaparib and durvalumab, that is a PD-L1 inhibitor, in patients with BRCA1, BRCA2, or PALB2 mutations that didn't progress to platinum-based chemotherapy. And we are waiting, we complete the recruitment, and we are waiting the results, and we will present probably the results next year at the ASCO GI meeting. ADCs, I want to only present 2 slides. This one is in order to see that we have a huge opportunities with the ADCs, because ADCs can be directed to different targets. And nowadays we have lots of clinical trials ongoing. We don't have a lot of clinical data yet, but we have a lot of clinical trials against EGFR, mesothelin, CEA, TROP2, tissue factor, ER3, so different targets in which we directed this ADC with different payloads, also different kind of chemotherapy insights, and we will see in the next year probably a lot of clinical data related with these specific drugs. I only want to present this data because it was presented this last ASCO meeting, and this is this drug that is an ADC specifically directed to CEACAM5 and 6, and in this phase 1 clinical trial, again in very refractory population, they demonstrated an overall response rate of 20% really, and a lot of those patients are really pretreated with some chemotherapy included in the pilot. There was a phase 2 that is planned in monotherapy, or also trying to combine with other chemotherapy strategies that it's ongoing. So let's see how this drug will move forward and the results in the probably next year. And with this, I want to thank all our team for all the work that we are doing against this disease, and all of you for your attention. Thank you.
Thanks very much, Teresa. Very detailed and very exhaustive revision. So we are moving forward in clinical trials. This is the aspect that we need for our patients. And any question from the audience? So we do need to study from the genetic point of view our patients, right?
Yeah, what we try is to avoid the situation in which we run a phase 3 clinical trial with 1,000 patients, even without tissue at baseline, no? Because then it's very difficult to understand which patients are benefiting from this or not, and then we have a negative clinical trial, no? Because not all the patients, of course, are homogeneous. So we try to convince the sponsors or whatever, so who run the trials, that we need biomarkers, because probably not all the treatments works for all the patients, no? But it's not easy, and always tissue in pancreatic cancer is so difficult to have for you.
Thanks. Ignacio?
Yeah, great review, Teresa. We have 2 studies from ASCO this year, very surprising. Elraglusib, it's a study in the metastatic setting that improves overall survival without improving PFS. We usually find the opposite, improvement in PFS that doesn't translate into overall survival. And then we have the PANOVA, a study testing a local approach that doesn't improve any of the local endpoints, but improves overall survival. It makes even less sense. We can attribute the benefit to immunotherapy. I guess that's what they are saying, right? That it caused some immune effect. But do you see anything on the elraglusib study that makes you suspicious that there might be imbalances? Like, for instance, they present patient characteristics. They look good, like they look comparable. But there's quite asymmetrical dropout in one arm versus the other that may have enriched one of the arms in a patient population with different characteristics. Every time I see something like this, that the treatment doesn't impact PFS, but impacts overall survival, I'm looking at what else we are missing before we go to a phase 3, enroll 500 patients to just get a negative trial.
Absolutely. I think we have to be cautious, really cautious, because we have really so many negative experience moving to phase 2. For me, when you do not have any impact in PFS, what's doing the drug here, it's very difficult to interpret. No? Then they presented the data of second line, or the percentage of patients moving to second line, and it was more or less the same. So with the presentation, the data that they presented, and we have to wait for the publication, but it was not huge difference. But for me, it's difficult to understand the impact in overall survival when you do not have any impact in PFS. It's not that it's not statistically no impact. it's difficult, and the difference in response rate also is so similar. And then I think TTFields is different. I think it's different because it's locally advanced pancreatic cancer. So there's an impact in overall survival. Believe it or not, there's 2 months of difference between both arms, and it's interesting to see how you have a difference in the metastatic, so in the distant metastases. The data is there. So why this is like this, we can think that maybe we can hypothesize. We don't really know. No? Local treatment, the local progression for me can be because it's very difficult to measure the local disease in pancreatic cancer. Maybe this can influence, and that's the reason why we do not have a local event. But yeah, for me it's provocative results. I don't know if, it's locally advanced pancreatic cancer, very, very difficult setting. No? Also, the endpoints are really difficult to measure.
Okay. Thanks a lot, Teresa. Congratulations for this excellent talk. Now we are going to have our last talk by Ignacio Garrido Laguna, who is presently the Professor of GI Oncology and Director of Phase 1 Program at the University of Utah at Salt Lake City, and he's going to talk about targeting KRAS beyond G12C.
I saw some people struggling before with this one, so I was trying to test how it works, but I guess we'll go on the fly. Thanks for having me here. Really appreciate the opportunity. It's really exciting always to come back home, right, and share some time with all of you. These are my disclosures. I'm going to be talking about a number of studies that are investigational. I'm active in the phase 1 space, so I have many of these molecules in clinical trials, in phase 1 trials, and I want to make clear that what I'm highlighting here, I'm not recommending the use of any of these drugs beyond a clinical trial, right? So these drugs are undergoing clinical testing right now. I'm going to start with the last, right? We have a myriad of RAS inhibitors that are now in the pipeline. It's a very exciting time for all of us working with pancreatic cancer patients because we have had really very limited treatment options to treat these patients. And for the last few years, we're seeing a change in the field. We know we can have an impact in the life of many of these patients. I think with what I will show today, there's plenty of evidence. We have proven the concept that KRAS beyond G12C is actionable now. And then, of course, combination therapy. I will show some heat maps or some maps showing some mechanisms of resistance. It's very clear resistance is polyclonal. It's going to be very difficult to target this disease with a combination of targeted therapy. I was very excited when I saw the data from Dr. Barbas's group today. Absolutely fantastic to see those tumor regressions and to see the durability of the responses. But we may need to still do combinations with cytotoxics or immunotherapy to be able to have sustained responses. This is, I think, we saw Silver presented a similar slide, right? I just want to acknowledge that it has taken 40 years to get here, right? And when I say 40 years, I see a lot of young people in the audience, a little bit less now because it's nearly 7 p.m., so I guess we had a few drops out, right? But look at this, we're here in 1982. I was 7 at that time, so it's really humbling to be talking about RAS inhibitors to the investigator who cloned the first oncogene, right? So I'm not going to go through this because obviously it's a slide that you will be able to see later and we have been discussing, but I think it's fair to make a point about, Kevin, really this doesn't point the direction I want, so I guess that's why people was struggling. But anyway, Kevin Shocker's lab was really the one who provided us evidence that this is actionable by identifying this highly reactive cysteine amino acid within the switch 2 pocket, a pocket that was not apparent. It's a pocket that was hidden and only exposed when the drug was present. So this is really what led or fueled the development of all the G12C inhibitors that you have seen lately, of course, with those 2 approvals that we have been mentioning. I'm not going to be talking much more about G12C, just give you a couple of hints. These are the 2 G12C inhibitors. They are not exactly the same. They are not really me-too molecules. You can see here how mutations in histidine in position 95 will alter or will not prevent or allow the binding of adagrasib. Adagrasib is a molecule that needs to interact with a few more amino acids than sotorasib, and that's why it's not able to disrupt the assembly of this complex RAS-RAF. But when you look at the data, you look at response rate, PFS, for the most is comparable, okay, between these G12C inhibitors. What else did we learn from G12C inhibitors? Mechanisms of resistance, right? So there's a number of other mutations that will occur on switch to pocket or new emerging escape mutations in KRAS, whether it's a G12D, a G12V, etc. Of course, mutations in other RAS genes like HRAS or NRAS, and then of course BRAF mutation. I want you to keep this on mind when I talk about the mechanisms of resistance to the pan-RAS inhibitor, because you will see how we don't see a number of these on that heat map. And more important than that, all this data is data that has been collected based on liquid biopsies. So we do cell-free DNA, and we are understanding the genetic mechanisms of resistance, but we're clueless about the non-genetic mechanisms of resistance. That's why we have this, more than half of the patients, we don't know what is the driving mechanism of resistance, probably because it's a non-genetic mechanism of resistance, whether it's activation of the, or loss of the hypo, and then activation of the JAP pathway, or adaptive resistance that we see emerge with this therapy. Second generation G12C inhibitors, and I promise this is my last slide about G12C. So very different molecules that have lower IC50 in vitro, lower dose in vivo, so more potent molecules. I'm not showing here, actually I'm showing divarasib here, which is a molecule that has more potency, but there's other G12C inhibitors that I'm not showing here that have a faster target engagement. It takes minutes for them to engage with G12C, while it takes longer for sotorasib and adagrasib. For the most, the response rate that we're going to see is going to be very similar. When you look at some of these studies that have a higher response rate, we need to be cautious because some of them didn't use Beaker, meaning the response were assessed by investigators. And we know when an investigator is assessing response, we tend to overestimate the response rate. And then, of course, the glecirasib study, but this was a China-only study, and there's still some concerns with responses in, let's say, China, just because the quality of the assessment perhaps is not the same as some of the Western countries. Moving now to, this is how the non-G12C landscape is looking. This is Shibuya. Some of you maybe have been there, right? Pretty busy area. And pretty much, I don't want to make this a boring slide. I want to give you some hints so that you can understand some of these drugs. We have, on the one hand, the multi-RAS inhibitors, the molecular glues. The one that everybody talks about, obviously, is 6236, the compound from Revolution Medicine. This is the first one, the one that has recently completed a randomized phase 3 trial in second-line metastatic pancreatic cancer. We expect sometime next year we'll have results from this study. But there's other molecules. So these molecules are being copied very quickly in China. Erasca has a molecular glue that just recently started a phase 1 first-in-human study in US. Same with GenFleet, and same with other companies that I'm not including here that are about to enter the clinic. I have bolded the RAS inhibitors that we have open at our center. Moving now to the second block, the pan-KRAS. Although they call them pan-KRAS, many of these molecules don't target G12R, which is pretty important for pancreatic cancer. It's present in around 20% of our pancreatic cancer patients. It's an alteration that it's hard to target. The reason is that many of these agents are targeting the off form of the protein, the GDP-bound form of the protein, and G12R essentially locks the protein in this GTP state. Going to the allele specific, what you can see here is there's a lot of G12D inhibitors, not that many G12V inhibitors. So we have this 3544, which is the first allele-specific inhibitor in the clinic. We have this open at our center. The reason is that this aspartate, it's harder to target, right? So the valine is harder to target. It's what biochemistry consider like a less reactive amino acid, so it's harder to develop compounds that target G12V. Even more important than this is what we have in the clinic now, but most of these, except for 375, are off inhibitors. Again, they target on the off form of the protein. We are going to have the first covalent inhibitor in the clinic. It's going to be probably less than 2-3 months. And I think that class of agents may also change or have a greater impact than what we are seeing with some of these off inhibitors. And I will be talking also today about some products. The first product for which we have results available is this 3082, presented at ESMO last year. We just opened the Arvinas molecule. There's a second product that it's open, not in our center, also from Astellas. And we just recently opened this, or actually we have the site initiation visit next week with this Arvinas molecule, which is another product targeting G12D. Now, I'm going to review some of the clinical data that you have seen presented recently. Some of the clinical data I will review has not been peer reviewed. I want to make that very clear. It's public data because the company disclosed the data in an investor conference, so you can all go and see the slides. But we have to be very careful with that data because it hasn't been peer reviewed. That means that the company can present the data whichever way they want, right? And then you have to interpret the data. Justo mentioned about the mRNA vaccines. I have a few slides, but I thought the presentation was going to be too long, so I took those, so I will just make a brief comment. There's 2 vaccines that have been tested on the adjuvant or neoadjuvant, actually on the adjuvant setting, the Eli 002. This is an off-the-shelf vaccine targeting G12R and G12D that has now been expanded to target other peptides, other KRAS peptides. The vaccine has been tested in the adjuvant settings in patients with high risk of relapse. These are patients with positive minimal residual disease after the Whipple, so patients that are expected to relapse within 4 to 6 months by CAT scan having a positive cell-free DNA. What they show with that vaccine, which is already published, is that patients who had response to the vaccine, immune response to the vaccine, had longer relapse-free survival compared to those who didn't respond to the vaccine. So it's a nice proof of concept. The second vaccine that you can see here, autogene cevumeran, it's a different concept. This is the messenger RNA vaccine that Justo was mentioning to you earlier, and it's a vaccine that it's customized. So the vaccine is developed based on the genomic profile, the neoantigens that those patients had at the time of resection, at the time they had the Whipple. What that means is that you had your Whipple, then you have to wait 5 months, right, while they develop the vaccine before they start giving you the vaccine and the immunotherapy. During that period of time, patients relapse. And in that study, we had close to 20% patients that relapse. So they don't really many patients won't get the opportunity to see the benefit from that vaccine. Why this particular vaccine was very interesting too, is because they had this case, when you read the paper, of a patient who developed a liver lesion while the vaccine, or shortly after the first dose of the vaccine was delivered, and then that liver lesion was biopsy. They did not find cancer, but they found atypical cells, so we have to be very suspicious that there were cancer cells there, and a high infiltrate of T cells. And that lesion eventually resolved. Why I'm mentioning this? Because for many years we have been saying vaccines in pancreatic cancer in the setting of metastatic disease are going to be useless because those patients progress very rapidly and they are better used in an adjuvant or setting. Well, that study provides proof of concept that we may see responses even in the setting of established metastatic disease. So 6236. So this is a molecular group, probably the drug that is raising the highest level of interest in our field in the last 2-3 years. I wish I had a plot to show you better how it works, but it's a glue that brings together two proteins that don't talk to each other or don't interact between themselves. So one is cyclophilin, the other one is RAS. When it does that, it causes some changes in the conformation of cyclophilin, so cyclophilin contributes to cause what they call steric occlusion, meaning the downstream signal mediators, RAF, PI3 kinase, are not able to bind activated KRAS anymore. So we did dose escalation studies, starting with low doses, 10 milligrams, escalating all the way to the 400 milligrams, which was not very well tolerated. So we end up staying on a dose of 300 milligrams for pancreatic cancer and 200 milligrams for non-small cell lung cancer, or what was the recommended phase 2 and phase 3 dose. This is data we presented at ASCO GI earlier this year, and I want to bring to your attention two aspects here. Look at the first line that those patients have got, FOLFIRINOX in around 75% of the patients. That's not really a good representation of what we see in the clinic. Around 40% of our patients are really fit to receive FOLFIRINOX in the first line. So when you see something like this, it speaks to patient selection. Same with number of percentage of metastasis at baseline, 68%. When we look at first-line studies, large randomized studies, we see around 80 to 85% of patients with liver mets. This is what happened with phase 1 trials. We get the supermans of oncology. We get patients who have gone through multiple lines of therapy, and they are still fit to get into a phase 1 trial. So it's always a little bit of perhaps not super representative of the general population of pancreatic cancer patients. So things that you need to keep in mind from this drug, even if it's targeted therapy, even if it's targeting an alteration, the drug, a mutation, the drug is also active against RAS wild type, right? And it's also active against HRAS and NRAS. So we do see a high rate of skin toxicity, skin RAS. So around 90% of these patients will develop skin RAS, very common. It is true the grade 3 skin RAS is not very high, staying less than 10%, around 8% or 7%, but we do see other toxicity, especially GI toxicity. We see quite a bit of nausea, vomiting, diarrhea, mucositis, right? So you will see diarrhea in around 50% of patients, mucositis, nausea, vomiting in around 30 plus 40% of the patients. I'm saying all this because something we don't know yet well is, even if we have done a phase 3 study in pancreatic cancer, do we have the right dose of this drug? This is very important these days, and a topic that is actively discussed in drug development in the phase 1 world. You may have heard of the Optimus project from the FDA. So what does that mean? When we're doing phase 1 trials, we need to choose the dose of the drug that provides the highest level of activity with a good balance of toxicities. What that means is that we look at the PK data, and when you see the PK data, this is doses and this is AUC, and you can see that there's not a lot of difference in exposure between the 200 and the 300 milligrams. It seems that exposure starts plateauing when you get at that level. So what do we need to look at? With similar exposures, you need to look at responses. Is this big enough of increasing exposure giving you higher response rate? We don't know that. The company has not shared that data. Is this higher dose a dose that doesn't lead to substantial increases in toxicities? We haven't seen data comparing the toxicity profile from 300 versus 200. So when you have a drug that causes, let me go back here, that cause those interruptions in close to half of the patient with those modifications in a third of the patient, you need to start thinking, do we have the right dose of that drug? And I just hope that we have the right dose of the drug, but I would like to see the data that the company used to choose the 300 versus 200 in pancreatic cancer patients. This is data they just disclosed last week, as I said, not on a conference, not peer-reviewed data, but it's probably the best data I have seen in pancreatic cancer second, third line setting in my entire career. we're looking at patients who receive this drug in the second-line setting. That's what I mean by 2L. By the way, I see a lot of young people here, and I don't know anyone understands what a waterfall plot, so I'm going to just comment very briefly for those of you who are not medical oncologists. What these lines pointing down are trying to show is the change in the size of the target lesions as we assess by RECIST. So when we're in the clinic, we do a CAT scan, we select 2 target lesions per organ, 2 target lesions in the liver, then we look at that CAT scan 2 months later and look at the percentage of decrease. So anything you see seen down is good. Anything you see going up is not good. It's tumor growing. This kind of waterfall plots in pancreatic cancer, this is unprecedented. A standard waterfall plot in pancreatic cancer, flip the waterfall plot and put all those bars pointing up. That's what we have seen for many years, and maybe 2 or 3 of those bars pointing down. Now, I want you to look at the PFS data and the overall survival data. It's impressive, right? PFS of 8.9 months with overall survival in excess of 13 months. In the second line, for comparison, the drug combination that we use as a standard of care, nalirify-FU, have PFS of 3 months and overall survival of 6 months. So we're doubling that. But look at what they did here, okay? They plotted this RAS G12X and then RAS mutant to create I have to say that this confused people, right? Because people see this graph and they say, oh, the RAS mutant do the G12 and the rest of the RAS do similar. No, they don't, because the yellow, which is the RAS mutant, you can see the curve is below, and what they did with this RAS mutant is not the non-G12. They included all the patients. So essentially, this is a Kaplan-Meier, whether instead of comparing A versus B being the non-G12, they are mixing together all the RAS mutations, and we know that the RAS mutant is driven by the G12. Of course, when you compare apples and apples, you get the same, right? So you're getting 70% of your group with the RAS mutant that are still driven by the G12. I don't think this analysis is meaningful. I think a more meaningful analysis will be to show you the outcomes of the G12 versus the non-G12. Why? Because we have some data from Pirolito showing that G12 mutations seem to respond better to the pan-RAS inhibitor, to the multi-RAS inhibitor. Why is that? Because it restores to some extent the GTPase activity of the protein. So the classic, it's hard to say classic when a drug is so new, right? But the well-known mechanism of action is that by causing this steric occlusion, activated RAS is not signaling through RAF or PI3 kinase. However, what he proposed is that in addition to that mechanism of action, for patients with mutations in codon 12, we do see some increase in the GTPase activity, sending RAS to the GDP-bound form, which is not active. So that is not seen with Q61. With Q61, you can see on that plot that the tri-complex is actually very stable and does not dissociate. So based on this data, we can expect that patients with mutations other than G12 perhaps will not do as well as the G12, but still will be better than chemotherapy because the bar is so low with chemotherapy. So that should not take much from the activity of the drug, especially because most of these patients anyway are going to have alterations in codon 12. This is the plot with the mechanisms of resistance that based on this study we presented at AACR this year. And what you can see here is that you continue to have all these mutations in the MAP kinase, but there's no KRAS mutations. Now what we see is a lot of KRAS amplification, consistent with what has been shown by Dr. Barbasit group and others with G12C inhibitors in this amplification of KRAS G12C. So the company has an allele-specific and a pan-RAS inhibitor. And obviously, there's a lot of excitement about combining both. And there's some preclinical data in PDX models that shows that they lead to synergistic activity, and the combo is superior to multi-RAS single agent. But when you have all these other mutations downstream that will not be targeted by the multi-RAS, and of course will not be targeted by the allele-specific, you need to think, is the combo really going to be a game changer for our patients? And I think that the combo may lead to perhaps a little bit more duration of activity, but eventually these patients are going to progress anyway. Data with first line. So again, this data was released by them last week, so not peer-reviewed, but this is really exciting data. So this is from the phase 1 trial. There was a cohort of patients that received the multi-RAS inhibitor prior to any other treatment. And what you can see here again, it's a response rate of 47%. This is impressive. The response rate, as you heard from Teresa earlier, to chemotherapy combinations range between 21 and 31%. We're talking about a single agent that causes a response rate that doubles the response rate that we typically see with chemotherapy. What they haven't shown us yet, it's what is the duration of response. Response by itself tells limited information, because if I have a patient that had a response on a CAT scan and the next CAT scan is progression, then that response is not going to be meaningful impact in the life of that patient. If I have a patient that have good duration of response, if I start seeing duration of response 7, 8, 9 months, then I'm start thinking this drug is going to impact life expectancy. Look at what I was mentioning earlier. Dose interruption in about half of the patient, 53%, dose reduction, 33%, dose discontinuation due to treatment-related adverse events, 10%. Someone asked me recently, why do you think this is higher than what they had reported in the second, third line? Because they didn't report those treatment discontinuation in the second, third line. We didn't see that. Well, maybe because these are first-line patients. When you are a second, third-line patient, you know this is it. Once you stop the treatment, there's no more. You go on supportive care. These patients first line, if they were having really miserable quality of life because of the mucositis, rash, or diarrhea, they may have a lower threshold to come out of trial, even if they're fighting pancreatic cancer. And then this is the data with the combination. So this is the multi-RAS inhibitor in combination with chemotherapy, nab-paclitaxel, gemcitabine. You can see response rate 55%. Things to comment on this slide, when we combine with chemo, one of the things that we look at are the side effects of the targeted therapy going to perhaps compromise the dosing with the chemotherapy. We don't really see that when it comes to dose reduction. 43% is very similar to what was seen in the clinical trials with this chemotherapy, but clearly the dose intensity was less than what was reported in the studies with chemotherapy, 75 to 80 down to 63%. Still, this is so much better than chemo, right? So I think there's no question that this is a combination that will need to be tested. And what is that they are planning to do? They are planning to do a randomized phase 3 study, so they are designing the study now, but what they are proposing is a control arm with nab-paclitaxel given per standard of care 1/8/15, which is how the MPACT protocol was developed. Then a combination arm with nab-paclitaxel given every 2 weeks in combination with the RAS inhibitor at a lower dose based on the safety study that has been done. And then once patients go to maintenance, they are increasing the dose of the RAS inhibitor to 300 milligrams, and then the experimental arm single agent. So 2 experimental arms compared to the control with chemotherapy. I think this is a very exciting study that may contribute to change how we treat patients with this disease in the short future, right? So short, because I expect the study is going to recruit very quickly. There's a lot of excitement with this drug. Now, going to the allele-specific G12D inhibitor. This is 9805. We have limited data with this molecule. This is coming from a phase 1 trial that was reported at the triple meeting by David Hong from MD Anderson last year. What I want to highlight here, look at the frequency of liver mets, okay, 86%. That's what we typically expect to see in these patients. Then they tested, of course, different dose levels. You can see those proportional increases in exposure, which is what we want to see in phase 1 trials. And then the question is, you have 2 drugs, an allele-specific and a multi-RAS inhibitor. This data, when it was presented, was to some extent considered disappointing because with an allele-specific inhibitor, the thought in the field was that the inhibition of RAS will be more potent and perhaps the response rate will be higher. It's not significantly different to what was seen with the multi-RAS inhibitor, but look at the toxicity profile. This is such a much cleaner molecule. You don't have RAS. You go from 90% RAS to 10% RAS. You have minimal or much lower diarrhea from 50% to 20%, and you don't see stomatitis. Patients are not complaining that it's hard for them to drink or to eat because of mucositis. When you look at the waterfall plots, a quick glance someone may say, well, you have a number of patients here that have growth that we don't see with the 6236, but here they are just showing you patients treated at the 300 milligrams. That was the dose that has been considered as the recommended phase 2 and the dose on phase 3. Here you have patients treated at all those levels. So if you get rid of the yellow color, you will get a picture that is actually not that different from the 6236, although we know, based on what I have presented you earlier, that the 6236 perhaps can contain the development of all these additional mutations in KRAS that an allele-specific inhibitor will not be able to prevent. This is a Chinese molecule that was presented at ASCO this year. It's a first-in-human study with perhaps the highest response rate reported to date in pancreatic cancer. We're talking about 50% response rate. It's the only G12D inhibitor, to my knowledge, that hits both on the GDP as well as the GTP-bound form. Things, again, look at the liver mets, 80%, more representative compared to the 6236 study of what we typically see in the clinic. And perhaps a little bit surprising, relatively better tolerated, which hard for me to understand because the rate of diarrhea is even higher than what was seen with the Revolution Medicine drug. One of the questions for the Revolution Medicine drug, we need more granularity on the GI toxicity. I presented that table that shows you the grade 3 toxicities and then the rest, but it's important to understand what is the percentage of grade 1, grade 2. Grade 2 diarrhea is not the same as grade 1. Patients may want to stop a drug for grade 2 diarrhea, while they may not necessarily want to do that for a grade 1 diarrhea. So all that information, hopefully the company will share with us soon. Look at the response rate, 52% response rate, fantastic waterfall plot. We don't really, the data is not mature yet to get to understand duration of responses, so we will need to wait for next presentation of this data in the upcoming months to see where these responses are durable or not. What are the questions about this study? This is a China-only study, okay? So we need to see patients from other countries being enrolled and see if the response rate holds. The response rate was assessed by investigators, so that's always something that we need to be careful because there's no central review of the images and there might be some overestimation of the response rate, and they included both confirmed and unconfirmed, similar to the data from Revolution Medicine. PROTACs. So PROTACs are small molecules, I guess, not exactly, but very close to a small molecule that have two domains, one that is going to be binding the protein of interest, and the other one is going to be binding an E3 ligase, and is going to be adding or tagging the protein to send it to degradation to the proteasome. This is the first PROTAC, to my knowledge, presented in pancreatic cancer, targeting G12D, presented at ESMO last year. Again, perhaps a little bit disappointing when you look at response rate, because the response rate was about 18%, while we already had data with the 6236 showing responses in the range of 30%. But this is proof of concept. To me, this is very exciting because it shows that we can drug RAS with a PROTAC, and the next molecule will be better than this, right? So a couple of comments, for instance, on DLTs. So these are dose-limiting toxicities. At 450, they did report a couple of DLTs with liver toxicities, which perhaps suggests that it might be challenging to combine this drug with immunotherapy, but too early to really know. We will want to see more data on this, and they have a better product that it's already on clinical trials, same company. So as I said, first in class is never best, or it's usually not best in class. This is just the beginning for PROTACs. Something I didn't mention here, when you look at the different dose levels, similar, again, with the caveat that these are small numbers, but similar response rate across dose level, even if we go higher. So this needs to make us think, are we starting to see a hook effect, right? Hook effect with PROTACs means that your drug is binding the protein of interest and it's binding the E3 ligase separately, but not assembling the ternary complex, right? So there's some sort of saturation, and it means that the dose response is not linear. It might follow like a bell shape, and maybe they are getting there, right? But again, it's too early to know based on this data. Strategies for combination. I wish I had shown you a KRAS, but I thought people will be showing a lot of these pictures of the pathway, and I haven't seen many today, but I already mentioned you have the GDP-bound form, the GTP-bound form. With G12C inhibitors, inhibiting upstream signaling from receptor tyrosine kinases is key. That's why we have seen increased activity leading to approval in colorectal in combination with cetuximab. With any G12D inhibitor that it's predominantly working on the GDP-bound form, it's going to be exactly the same, because you need to try to increase the pool of protein that it's in the GDP state. So you will see combinations with upstream inhibitors, EGFR, SHP2, not well tolerated, of course allele specific with the multi-RAS, and then CDK4/6, TEAD inhibitors, which are already available in the clinic, and these are being developed. But to me, to address polyclonal resistance, we're going to need non-targeted mechanisms of action. We're going to need combination with chemotherapy. We're going to need maybe combination with immunotherapy. So I have a slide, I think it's at the end, where I talk a little bit more about the classic and the basal phenotype as part of my conclusion, so I will wait to introduce that concept later. So co-targeting, this is actually colorectal models. This is not pancreatic cancer, but it's to give you a sense that this strategy is being developed or being evaluated. This is G12C in combination with SOS1. And at the triple meeting in a few weeks, we're going to present the first data with a G12D off inhibitor in combination with cetuximab, an EGFR inhibitor, following the concept that I just mentioned to you. Other combinations, this is from Andy Lawis' lab at UCSD in San Diego, looking at inhibition with RAS leads to increased expression of HER2 and JAP that may lead to increased kind of RAS-like signal. And you can see here how the combination increased the life of those mice. Of course, these mice die. They are not like Dr. Barbasit plot that was flat right here, right? We have heard about this before, so I'm not going to comment much on this, but this is a genetic alteration that is very common in pancreatic cancer. Around 20% of our patients have this MTAP loss. You heard Teresa mentioning the BMS study where patients were selected based on CDKN2 mutation or MTAP loss of function. And the response was modest because they don't always go together. So they are in kind of the same region, the same chromosome, but we do see sometimes patients who have CDKN2 mutations who don't have MTAP loss of function. So key here is to select patients who have homozygous MTAP loss of function. And when we do that, we see responses in the range of 20%. Again, this is just from a slide from the company, but there's multiple PRMT5 inhibitors. All of them, I will say, based on the data we have, leads to similar response rate in that range of 20%. This is a patient with, I think it was a Q61 mutation actually, who had already progressed to a RAS inhibitor, went into this phase 1 trial with TNG462, and had a nice partial response. So based on, you already heard this from Teresa, that we actually have this study about to open at our center, combining the PRMT5 inhibitor with either the multi-RAS inhibitor or an allele-specific RAS inhibitor. And this is data with FAC, right? This is a study that it's ongoing now, a phase 1 that we also have open at our center, combining a FAC inhibitor that we know leads to increased phospho-ERK with a RAF-MEK clamp. The data that I'm showing here shows synergistic activity in PDX models, and based on that, we are doing a phase 1 trial, and again, not peer-reviewed data, just released from the company, where you can see with a small number of patients, 10 patients with unconfirmed response out of 12 patients treated, with a rate of confirmed partial responses close to 67%, which is much better than what has been reported on phase 3 trials. With the caveat, this is a small number of patients. We tend to overestimate the response rate that we see in these small studies. So what's the future? I already give you a few clues. We're going to have the first covalent inhibitor targeting G12D in the clinic very soon. We're going to have better molecular glues, so Revolution Medicine might be ahead of the pack, but the competition is going to be fierce. There's molecules that have, based on preclinical models, lower IC50, lower dose in terms of milligram use on those mice model compared to what Revolution Medicine has been using. How are we going to address polyclonal resistance? Combination of multi-RAS with an allele specific, with all the caveats that I have discussed. Cytotoxics. In pancreatic cancer, we know that based on RNA sequencing studies, you have heard from multiple speakers, we have these two populations that we typically see mix in the tumors, but at different percentage, the basal, the classic. So there's work from Andy Aguirre from Dana-Farber showing that when you treat these models with a RAS inhibitor, you push towards the classical phenotype, because the basal is what responds better to the RAS inhibitor. So you can, with chemotherapy, is the opposite, right? We talk about patients with a classic phenotype responding or benefiting to a greter extent from chemotherapy compared to the basal phenotype. So you can imagine this combination, chemo plus a RAS inhibitor. Chemo is hitting on the classic phenotype, the RAS inhibitor is hitting on the basal. Same for combinations with, for instance, antibody-drug conjugates, right? For targeting Claudin 18, which is highly expressed in the classic versus the basal phenotype. Immunotherapy, I already mentioned why. Other targeted therapies, I already mentioned perhaps with the polyclonal resistance, these may have limited mileage. And then we didn't talk today about the non-genetic mechanisms of resistance. This is something that we need to understand better. And to understand this better, we need to collect biopsies on this trial. We need to be able to do RNA sequencing studies on patients at baseline, then at the time of progression, and dissect the non-genetic mechanism of resistance. I think I have a last slide. This is one of my patients on one of these RAS inhibitor studies. He has been very public about his disease, and that's why I'm showing his picture here. His name is Jerry Simonson. He went on this trial in 2022, November 2022. He went at a low dose, and I'm showing this to show you the benefit of going into a phase 1 study. Low dose is 80 milligrams. We said that the phase 3 trial has been done at 300 milligrams, right? Look at the amazing response he had. It's a patient that had only lung mets. We know those patients tend to do better compared to those that have liver mets, but these patients, sadly, still succumb to the disease. And he's 3 years out, no evidence of disease on a CAT scan, doing so well. And I don't know if someone can plot the little video I have. He's a former football player for Utah University. So there's a beautiful video of him. The link didn't work, so if they're not able to play, that's fine. But he's on the special teams. These are the teams that go when you kick the ball, right? And the video is him scoring a touchdown, his only touchdown, because special teams usually will not score a touchdown unless the other team fumbles the ball, which is exactly what happened here. So that's why I think this for him is really a touchdown, being 3 years out with no evidence of, oh, look at that. We were able to, thank you. So being 3 years out with no evidence of disease is impressive. Like, as a treating physician, you can imagine that the joy in our team every time we get to see him. And I think it's okay. I already told the story. So you're going to watch a touchdown, right? So it might not be something you see very often here in Spain. That's him running for the fumble, and he got it there, okay? Yeah. The fumble happened before, right? They dropped the ball. The fumble is when they drop the ball. And I think that might be it. There was a slide acknowledging our phase 1 team. I show some of them on the picture, but that's all I had for you today. I'm really thankful for you staying here this long. It's nearly 7, so.
Thanks very much, Ignacio. It's been a privilege hearing you with these new opportunities for our patients. Any suggestion or any question from the audience? Yeah, Nuria, please.
Thank you, Ignacio. Ignacio, super interesting talk and view of these complexities also. I wonder, what do you think, or is there anything already running regarding to apply vaccines, RAS vaccines, previously as a chemo prevention or immune prevention strategies among those individuals with premalignant mutated RAS lesions?
Yeah, I'm not involved in those studies because I mostly get to see our patients once they are diagnosed, right? So you're talking about patients who have panning lesions. We don't really see the panning lesions. There's a lot of studies looking at prevention with ultrasound, but that's still very early. There's a study published by Johns Hopkins like 2-3 years ago, and even when you diagnose these patients early, there's no early. Once you see the cancer on an ultrasound or on a CAT scan, that's already too late. Like there's a beautiful paper from, I'm blanking on Andrew Ream from 10-12 years ago, looking on a mouse model, and on that mouse model, when there were only panning lesions, they were able to see already cells that have already seeded the liver. They marked that, and they were able to see cells. So that tells you that there's some invasive component very, very early, even if they thought that they were only panning lesions. I think the opportunity will be for those other patients who have IPMN lesions, who have main duct IPMN lesions. We have a speaker talking earlier today, I think it was Mary Chell, talking about the difference in the profile of cells coming from the duct versus from the side branch, from the smaller ducts. And because those we can see on a CAT scan, and we know that they have a high risk of malignancy, the main duct, the IPMN, are lesions in the main duct that if we don't take that lesion out, 40% of the patients will develop invasive cancer. So these patients will typically do surgery different to the side branch IPMN that we can watch because they are less likely to lead to invasive cancer. But to your answer, I'm not an expert in vaccines in that field because I don't get to see patients in that setting. It will be more our GI doctors. I think it's even more important than what we do because when we get there, we are late already. So working on prevention, identifying those high-risk patients is key. What I was going to tell you is that on that study from Hopkins, okay, even if they were screening patients, those patients already had like stage 2 pancreatic cancer. Most of them, when they were diagnosed, most of these patients, when they went to surgery, had already positive lymph nodes. So even with the screening high-risk patients for pancreatic cancer, we got late because we took them to surgery when they had already positive lymph nodes. So I think we need to work a lot to refine who are high risk, maybe develop blood signatures that can help us identify who these patients are, right? And because by image it's really hard to identify them. Yeah.
Yeah. We have an hour behind the schedule, and I am going to make the last question. The phase 3 trial on the
236. Yeah.
236, yeah. Is it going to be international among in Europe also?
I cannot speak for them, but I'm pretty sure it's going to be a global study like the second-line study was.
The first studies were only developed at the States. And,
right. The phase 1 study was done mostly in US centers. The second-line study has been a global study that has accrued very, very well, very, very rapidly, and I will expect that the phase 3 will be also a global trial.
Now it's an opportunity for the patients.
Yeah.
We see that.
Yeah.
Yeah. Okay, thanks very much, Ignacio.
Thank you. Thanks for having me here. Muchas gracias por tenerme aquí. A ver si estamos en contacto. No, mucha suerte.
Okay, I have a duty that Mariano has told me that is the wrap-up, which means to resume in 15 minutes the 14 talks. So I have to say that I liked very much the first one with the acinar versus ductal origin of the pancreatic ductal adenocarcinoma by Marichel. And I also liked very much Bruno Sainz's talk about the cancer stem cells and the riboflavin inside and its IGN 111 trial. And also the attitude of Justo with the spliceosome and the SF3H1 and the neoepitopes that were available after splicing the genes. Paco talked to us about NR5A2 mice with the pre-inflammation related to the microbiome and these mouse models that included CTRB1 and 2 genetic modified. So TUTCA for PDAC prevention could be also an opportunity, and Paco has exposed this very brilliantly. Microenvironment and treatment resistance in PDAC mouse models were exposed by Jean Morton, and the combination of mTORC MEK inhibitors, RMC7977, was commented, as well as gremlin-1 inhibitors. Combined targeted therapy induced complete response with tamoxifen in EGFR and RAF1 and STAT3 inhibited mouse models from Mariano, Carmen, and Vasiliki that were brilliantly developed at their CNIO center. Now they are working with a promising agent RMC6236 plus an upstream inhibitor, afatinib, plus SD36 that has a good tolerance and a promising efficacy. After that, Peter Bailey talked to us about progression and resistance mechanism, the transcriptional states, the switching between the Basel-like type and classical one, the markers, GATA6, KRY17, and CYP3A phenotypes, the genetic MYC burden, which is important and lately was developed in the last table. And he also stated the importance of the extracellular DNA and chromosomes as resistant mechanism for therapy. Tumor host interactions in KRAS dosage variation and biology with the RMC6236 by Dieter Sauer was commented, as well as the subclonal architecture, evolution, and resistant mechanism to KRAS 12D. The microenvironment, the heterogeneous microenvironment, is difficult to target, and there is a lack of preclinical models. and reprogramming this microenvironment with TXCL5 was suggested. Neutrophils and macrophages towards T cells immunosuppression were also among their lines of research. Nuria Malatz spoke to us about diabetes and obesity in PDAC, a multifactorial disease, and she brilliantly exposed the PanGene-EU study with 2,500 people recruited, and the new onset diabetes, which increases the risk of PDAC by 6 times in the first 2 years, as well as men had 8 times the risk than women, am I right? Yeah. Were discussed together with the BMI. BMI, it's a thing that has to be considered together with type 2 diabetes, but before that we have to consider the lifestyle habits, the genome, the microbiome, the metabolome, the epigenome, and to check if diabetes and PDAC has a mediator or effect modifier relationship. NR5A2 in diabetes and PDAC are present, and methylone, diabetics, and smoking are already demonstrated. Integrative epidemiology model is progressing with Nuria. The next speaker was about resistance to PDAC treatment. Nelson Ducetti talked to us about targeted therapies, drug chemotyping in preclinical models, FOLFIRINOX and GEM, as Abraxane was not refounded in France, and they are working with Teresa Macarulla to check if nab-paclitaxel obeys their model. He has initiated a single-cell RNA-seq. He has also commented about intratumoral heterogeneity and the integration of the tumor microenvironment in the future model. This is Nelson Ducetti. The potentiation of KRAS inhibitors by Silvestre Vicente was the combination of single-cell technology, computational work, actionable mutations, and common central effectors with clinical relevance. Cross-tumors KRAS own signature were always in every species a bad prognostic factor. He was talking about FOSL1, LAMB3, and LAMC2, which are overexpressed in PDAC. These laminins are present in the desmoplastic stroma and even in the preneoplastic lesions like PANIN3. So they should be useful for early detection of pancreatic cancer. Experiments with LAMC2, with a combo of RMC 7977 and AXL are ongoing because they are synergistic. Laura Susek talked to us about MYC. And MYC is the most regulated gene and difficult to inhibit. They have developed omomic-derived mini proteins are pharmacological tools that display tropism for many tumors. They are like theragnostic. Theragnostic means that they can be useful for diagnosis and for treatment because they accumulate in the tumor. You see the extension of the tumor at low doses, and you increase the doses for therapy. It has good tolerance and efficacy. It can be injected intravenously, and they have a phase 1 trial already finished with one partial response in one pancreatic cancer, liquid biomarkers, and the phase 1b together with nab-paclitaxel plus gemcitabine has already finished its recruitment. The new options that Teresa developed are multiple, and she started with the targeted therapies, which are more useful for younger people and for familial cancer because they appear in a higher proportion in this population, like 30%, 40% of people younger than 50 years old and familial cancer do not have RAS mutations. So they can benefit from any other targeted therapy like selpercatinib, like zolbetuximab, which is an agent against Claudin 18.2. There are many other agents against Claudin 18.2, like EV343, EV389, which is B-specific and together combines Claudin 80.2 with CD3. And there is a trial named Clarity with an antibody drug conjugate that is going on. Another aspect that she commented was the MTAP deletion and the dependency of these patients on PRMT5 and the AMG 193 drug, as well as Bristol-Myers Squibb 96854 or TNG 482 are drugs that target these patients' currency. Another drug is elraglusib, a GSK3 beta inhibitor, and she commented on that as well as in immune therapy with CD40 agonists, plus FOLFIRINOX or nivolumab. Oncolytic viruses were also on the scenario, plus chemo, a CD73 inhibitor. The tumor treating fields also were commented. Now it's planned to tumor treating fields with chemo or immune therapy. And finally, she ended with the DDR alterations, BRCA1, 2, and PALB2, that benefit from olaparib or rucaparib that have been developed now as an adjuvant treatment. They were effective in the metastatic setting only in disease-free survival, not in overall survival, olaparib. And now there is also an antibody drug conjugate that targets these inhibitors with different payloads to check the efficacy and the tolerability. The last, but not the least, talk was given by Ignacio Garrido Laguna. And Ignacio has talked a lot about adagrasib and sotorasib, which are the first G12C inhibitors, and they can finally be ineffective because resistance is not genetic in more than half of the patients, in 54% of them. So there is now second generation of these compounds. RMC 6236 was commented also, as well as allele-specific inhibitors, PDAC inhibitors, vaccines like LE002 that was recently published in the adjuvant setting. Those who develop a T-cell response, they benefit from the vaccine and they don't recur. So it's important to check the immune status of the patients after the vaccine. And the RMC 6236 was explained also by Ignacio with a cyclophilin that occludes the pocket and inhibits RAS, and maybe overrated the dose of 300 milligrams of the drug, and it could work properly with 200 milligrams, which is the dose that is used in lung cancer, and RAS is more than 90%, grade 3 RAS. This is not recommendable, as well as diarrhea and nausea and vomiting. And in second line, this drug gives 35% of response rate and 8.9 progression-free survival months, which is incredible, and an overall survival of around 1 year, which is what we obtain with chemotherapy in first line. And in first line, the response rate with this drug is 47%. Dose discontinuation was only in 10% of patients, but the lowering of the drug dosage or the delay of the treatment was in 50% of the patients each. And there are other phase 3 trials coming, like the Resolute 303, that we hope that will come soon to Spain. Will test this drug in the way that Ignacio said. And he also talked about the PDAC targeting G12D and the proteasome that was in the beginning was a result that somewhat was not what everybody was expecting, but it's the beginning of a long way. Strategies for combo upstream, like EGFR inhibitors, CD4/6 inhibitors, TET inhibitors, etc., with this drug. And the future is new covalent inhibitor beta-glues, also, and combos with allele-specific plus pan-KRAS inhibitors, etc., etc. So I would like to thank you for staying here until the end. I would like to thank Carmen, Mariano, the CNIO, for organizing this. I would like to thank also the Fundación Ramón Areces for allocating us here, and I am happy to be at this time of the evening together with you enjoying this time. Thanks a lot.